The Royal Swedish Academy of Sciences has today decided to award the 2020 Nobel Prize in Physics with one half to Roger Penrose for the discovery that black hole formation is a robust prediction of the general theory of relativity.
Welcome back to day 3 of the Science and Roger Penrose live webinar. Congratulations, Roger, on your Nobel Prize and happy birthday. It's a pleasure and an honor to be with you today as your session chair. My name is Justin Riddle. I'm a cognitive neuroscientist and postdoc at the University of North Carolina at Chapel Hill. I use non-invasive brain stimulation in humans to modulate neural oscillations, and I've taught an undergraduate course on quantum mechanical theories of consciousness. It's an honor to be here with Roger Penrose himself. On a personal note, I found a profound source of inspiration from Roger's work, most notably his 3-world model of consciousness and the orchestrated objective reduction model, which you'll be hearing about today. So in the past 2 days, we've done a deep dive into the physics inspired by Roger's work. And in today's plenary talks and panel discussion, we will explore how the principles of quantum mechanics are utilized by biological systems. Is your brain a quantum computer? Do microtubules and neurons orchestrate macroscopic superpositions into a series of conscious moments via objective reduction? To kick off our session entitled Consciousness, Orchestrated Objective Reduction, we will be hearing all about the Orch OR theory of consciousness from Dr. Stuart Hameroff. Dr. Hameroff is a clinical anesthesiologist at the University of Arizona. He is a co-founder and current director of the Center for Consciousness Studies, and he organizes the annual Science of Consciousness Conference. After proposing a model by which microtubules instantiate a flexible universal digital computer at the core of neural function, Stuart teamed up with Sir Roger Penrose and updated his microtubule model to be a quantum computer. Stuart provides the biological complement in the Penrose-Hameroff dynamic duo, and since its inception, the Orch OR model has been fine-tuned and extended. At present, multiple research grants and laboratories are investigating the validity of the Orch OR model. And on a personal note, Stuart and Roger have been an inspiration for me in their bravery to develop a novel and compelling interdisciplinary theory that seeks not only to solve the mystery of how consciousness arises from neural activity, but simultaneously solve fundamental mysteries in quantum physics. So without further ado, let's hear it from the pioneer himself. Welcome to the mic, Stuart Hameroff.
Thank you, Justin. Thank you all for being here. Thanks to the organizers for inviting me. That's me, of course. And thanks to Roger for starting all this. I'm going to talk about the Orch OR theory of consciousness. And on my first slide, so here we see, here we were at the Grand Canyon in 1994. Roger's in the yellow shirt. I'm just behind him. On the far left is Dave Chalmers, a severe critic of ours, but also a very good friend. So this is when we began to actually talk seriously about what we wanted to propose. And so let me start with Western philosophy and neuroscience, Plato or somebody like that, who recognized that the world out there is all in our head, being. And I will use being to connote subjective phenomenal experience, consciousness, the difference between doing something on autopilot and doing something consciously. The hard problem, the explanatory gap, whatever you want to call it, but it connotes conscious experience as opposed to just cognition without consciousness. So in the West, it's all in our head. Being connotes consciousness. In Eastern philosophy, consciousness pervades a deeper level of reality. Being is everywhere. We are basically a wave in a sea of being, you might say, according to Eastern philosophy. Now, in the brain, we know that conscious perception involves 3 waves from thalamus. The thalamus is in the middle here, and sensory input from the eyes here, from the spinal cord, the ears, all come into the thalamus, all sensory modalities except for smell. And there are 3 waves from the thalamus for primary cortex. For vision, for example, goes to the back of the brain, and then from the V1, the back of the brain, the second wave, integration, shape, color, motion, meaning, goes to the front of the brain and to the prefrontal and frontal cortex. And then a third wave from prefrontal and frontal cortex. The third wave goes to other areas of cortex and other areas, and consciousness occurs in the third wave. And we know that because anesthesia only affects the third wave. But it occurs 300 to 500 milliseconds after sensory input, Bing. That's when Bing occurs. And even though we often respond consciously within 100 milliseconds. This is a problem because consciousness is therefore considered epiphenomenal and illusory. We respond non-consciously and have this false illusion after the fact that we responded consciously. That's pretty much the conventional wisdom in neuroscience, because the activity correlated with consciousness appears to occur too late. Roger's physics actually has a way out of this that can rescue conscious free will, and we'll come back to that. From these areas going into the cortex, from thalamus or other cortical areas, signals also progress in 3 waves, first to layer 4, then from 4 to 1, 2, 3, and 6, and then converging on layer 5. Layer 5, giant pyramidal neurons are I would say, the most likely site for consciousness. Carl Pribram in 1972 pointed out that the basilar dendrites that connect the pyramidal neurons this way form a web, a dendritic web that would produce interference, and what he proposed was a holographic model of consciousness. But I think that the pyramidal neurons are the most likely site for consciousness, according to our theory and other theories. Now, if we look at the giant pyramidal neuron, we see the conical or pyramidal-shaped cell body. The apical dendrite goes to the surface of the cortex, gives rise to EEG. The basilar dendrites go laterally to form the web, and the axon goes downward directly to the spinal cord, to the pyramidal tracts. And we see one particular microtubule made up of individual tubulin proteins inside the neuron. And what Roger and I are proposing is that consciousness extends downward inside neurons to the level of microtubules, in particular to quantum effects in microtubules. If we look a little bit closer at the pyramidal cells, the microtubules are actually in mixed polarity networks where the polarity is opposite in adjacent ones, and we think that may promote interference patterns. The dendritic somatic microtubules are short and interrupted. We also see the centrioles, which are found in all cells, but are particularly interesting in neurons. And finally, below, we see that firing starts at the axon initiation segment where microtubule arrangement changes and motor control takes over. So the axon is defined by the arrangement of the microtubules, not just the appendage heading downward. The proximal axon is actually acting like a dendrite, and it changes at the AIS. And here we see an immunomicrograph of 2 pyramidal neurons, and those yellow things are the cilia, and you see those barrels, the centrioles and centrosomes and centrioles in the middle, those barrels. One of them extends outward and acts as an antenna, which can detect photons actually in the extracellular space. And neurons have extremely well-developed primary cilia, they're called, that extend outward. And they remain a mystery in neuroscience. Now, the neurons themselves are thought to behave as fairly simple Hodgkin-Huxley integrate and fire neurons. We have cell body and dendrites that receive and integrate neurons. And here's the axon. At some point, this will get triggered and fire a spike or an action potential down to the next neuron. And this is all mediated, according to Hodgkin-Huxley, by ion channels opening and closing on the membrane surface. So it doesn't take into consideration anything in the neuron. It's all inside the neuron. It's all based on ion channels and membrane propagation. And so integration in the dendrites and soma, firing in the axon, integrate, fire. And this is the conventional view of what neurons do. And the schematic for this is shown here, where the integration occurs here and the firing occurs here, and it's a fairly narrow threshold. And on this axis, we can see a narrow temporal window and a narrow voltage threshold to get a very narrow spike. And it's sloped here because the because the ion channels are supposed to open sequentially. So we get this sloping action potential. So this is the conventional picture of what is supposedly going on with the Hodgkin-Huxley neuron. This would suggest the brain is a computer of algorithmic Hodgkin-Huxley integrate and fire neurons. On the left, we see a tangle of neurons. On the right, we see a bunch of nodes in a computer switching matrix. And this type of completely algorithmic behavior leaves no place for consciousness, creativity, intuition, insight, or free will for other reasons because of the time. But fortunately, there's hope because, and that of course would be consistent with the idea that consciousness is epiphenomenal and illusory. However, if we look at the recordings from an actual neuron, a pyramidal neuron in an awake cat from this study by Nondorf et al. From 2006, we see a very wide threshold for firing. So we have a wide voltage threshold and a wide temporal variability, and the spikes are vertical because it turns out that these ion channels in this area open simultaneously, that they might actually be quantum coherently coupled or something like that. But in any case, they act cooperatively, and we get this vertical spike, and it's very broad because of temporal variability and wide threshold. So this is where some non-computable factor, and Roger, of course, brought up non-computability as an important component of consciousness in his book, The Emperor's New Mind. Some non-computable factor regulates the firings of the spikes of the neuron and the behavior of the neuron. And this is where consciousness could come in and regulate behavior, supervene on non-conscious cognitive function. And so here we see the Bing in where it might be regulating the firing of the axon. And where is this coming from? Well, it can't come from outside because then we'd see it in the membrane potential. So it must be coming from a deeper level. For example, inside the dendrite here. So here's the dendrite and the cell body, and here's the axon where the firing is going to occur. And it likely to be coming from inside in the microtubules inside the dendrites and the soma, the cytoplasm and the cytoskeleton. And so Bing may actually be in consciousness in the dendrites and soma prior to causing triggering the spike. This is somewhat similar to deep learning networks or convolutional networks in artificial intelligence, although they're interspersed serially, but they could be at a deeper level inside, if that were possible in a computer. So this kind of lends itself to the Orch OR dendritic, the Orch OR orchestrate and fire non-computable neuron that we see on the right, which has a lot of microtubules and stuff going on that could modulate and supervene on the membrane activities and bring consciousness into the picture and trigger conscious activity. Now, we know a lot about microtubules. For example, in Alzheimer's disease, the microtubules fall apart. If your microtubules get destabilized and the tau, the little blue squiggly things, fall off, the microtubules disassemble and disintegrate, and you get Alzheimer's. And whether the tau falls off first and that destabilizes, or the microtubules destabilize and the tau falls off, in either case, the microtubules are disassembled, the neurons shrink, you lose synapses. And even though the amyloid plaques get all the attention, what causes the cognitive deficit is the disintegrating microtubules and loss of tau. Now, tau serves a role as kind of a traffic signal along the microtubules, which serve a lot of functions, including transport. So materials that are needed out at a synapse here are synthesized in the cell body here and are transported along a microtubule by these motor proteins, which move along them and are triggered or signaled by the tau proteins when to get off and deliver their cargo to a particular synapse. So this is synaptic plasticity as it actually occurs. And so if the microtubules disassemble or the tau falls off, you lose this traffic signal and you lose synaptic plasticity. Microtubules also probably serve as a site for memory, as shown in this study from Travis Craddock and J.Tuszyn and others, where the CAMK2 molecule can phosphorylate up to 6 bits of information on a microtubule. The lattice structure and the organizational abilities of microtubules have long prompted the idea that they serve as an information processing capacity. And going back to the early 80s, when Rich Watt and I published in the Journal of Theoretical Biology, and going forth from there with Jack Tuszyn, Steen Rasmussen, Travis Craddock, and a number of other colleagues, Aarat, Kalra most recently, and others, looking at microtubules as information processing devices, and then beginning with Roger, the idea that microtubules act actually as quantum computers. So one aspect of this is that there are a lot more tubulins in the brain than neurons. And if you want to calculate the brain's computational capacity at the level of neurons as fundamental units, which most people do, the AI singularity, there are 10 to the 11th neurons in the brain, about 1,000 synapses per neuron at about 100 hertz, gives 10 to the 16th operations per second per brain. If you consider the microtubules, the same 10 to the 11th neurons, 10 to the 9th or 10 to the 8th tubulins per neuron, oscillating at about 10 megahertz, and I'll explain that a little bit later, you get 10 to the 16th operations per second per neuron, or 10 to the 27th operations per second per brain, which is a lot, but it's still algorithmic. There's no provision for consciousness. Its computation is something else required. And this is the essence of the hard problem. What is required for consciousness? So here's a very quick overview of the philosophy of mind and the hard problem as I see it. Panpsychism says that mind is essentially the same as matter, or that material has aspects of mind associated with it. So they're more or less equivalent. Emergence materialism, computationalism suggests that matter generates mind. Idealism, or the Copenhagen interpretation, where consciousness causes collapse, says that mind creates or modifies matter. Dualism would say that mind and matter are separate. And neutral monism says that there's something, this is Bertrand Russell, and this has been popularized by Harold Ottmanspacher, that something vast gives rise to both mind and matter. And this can be, this is equivalent, I think, and is consistent with the Orch OR theory, and I'll explain this a little bit more, that quantum space-time and brain microtubules gives rise to both mind and matter when collapse or objective reduction occurs. So is consciousness truly computation? I read Roger's book in the early 1990s and was really impressed by it for many reasons. To summarize, through Gödel's theorem, Rogers argued that consciousness is not algorithmic and therefore not computable. A factor outside classical computation was required for mathematical proof and for conscious understanding, something non-computable. And he saw a potential source of non-computability in the unexplained quantum state reduction or collapse of the wave function. And he proposed this was due to an objective threshold related to quantum gravity or to fundamental space-time geometry, providing an objective threshold, therefore objective reduction or OR. So to delve into the quantum world, not being a physicist, I'm going to take liberties and present it the way I kind of like to understand it, that the world is divided into two realms, the quantum and classical realm, which have different laws. And in the quantum realm, we have superposition of multiple coexisting possibilities. Things tend to be small, non-local, and wave-like. And in the classical world, things are localized, particle-like, and large. And OR, you might say, exists on this edge between the quantum and classical worlds, as does consciousness, kind of on this edge between the two worlds. In quantum superposition, a particle can exist as a wave of multiple possibilities. For example, here's a cesium ion as a wave in the background, or in the foreground as a particle in a definite state and location, classical. When quantum waves are measured or observed, they become particles, or they appear to become particles in definite states. This is the measurement problem. So if you looked at if you could attempt to look at the waves by looking or by measuring, that would cause the waves to collapse to a single atom in a particular location. This led Schrodinger to recognize the absurdity that the observation caused collapse, and he came up with this famous thought experiment of Schrodinger's cat and quantum superposition of both dead and alive until observed by conscious entity, presumably human. Wigner, von Neumann, Stapp, more recently Chalmers and McQueen have taken this view. And the idea is that consciousness causes collapse, putting consciousness outside science. So yesterday, Philip Stamp said that I think he was referring to this view was medieval and irrelevant. And from the standpoint of this approach, that consciousness causes collapse, I would agree with him. But let's first ask the question that Roger did: how can a particle actually be in multiple locations simultaneously? And he accounted for this through he accounted for superposition through Einstein's general relativity, which linked matter to curvature in fundamental space-time geometry. And for example, a star behind the sun, the light would be curved, so we could see it even behind the sun. And this is Eddington's famous experiment that proves Einstein's theory that space-time curved. That's for large objects. What about matter and space-time curvature at small scales? Roger used these two-dimensional space-time sheets with one-dimensional space and one dimension of time with a particle equivalent to a curvature. And a curve in one place is a particle in one location, and another is the particle in a different. And they can oscillate back and forth. So a particle oscillating between two positions would be the space-time curvature oscillating back and forth. And superposition, quantum superposition, would be opposing curvatures, separations in fundamental space-time geometry, shown at the right. So this forms kind of a qubit, a space-time qubit, if you will. Now, you could imagine if these separations were to continue, that we would get multiple worlds, the idea that there is no collapse, that every possibility persists and creates its own world, its own universe. So here we have two curvatures going their separate ways, forming their own universe. And of course, Roger would say those separations are unstable. We'll come back to that. So here's the view that consciousness collapses the wave function I mentioned before. And I call this subjective reduction as opposed to Roger's objective reduction. In this view, the being is in the conscious observer, but we're not explaining being. We're not explaining why he or she is conscious, nor are we explaining why conscious observation collapses the wave function, nor what what it actually means to be in superposition of multiple possibilities. So I would disagree with this approach. It's dualist. It puts consciousness outside of science. Roger said, however, that superposition separation is unstable, will self-collapse, and undergo objective reduction, OR, at a time t equals h bar over E sub g. H bar is Planck's constant over 2 pi, or the Planck-Dirac constant. E sub g is the gravitational self-energy required to separate something from itself or separate space-time from itself. And this was the kicker for sure, when that happened, there would be a moment of proto-conscious experience, or moment of consciousness, Bing, emanating from this collapse derived all the way down from the Planck scale, space-time geometry. So rather than consciousness causing collapse, OR causes or is equivalent to consciousness. So just to drive this point home one more time, on the top we see subjective reduction, conscious observation causing collapse. The bottom we see objective reduction, collapse causing consciousness or being equivalent to consciousness. In the random environment, such OR events would be isolated and lack meaning and context. They would be proto-conscious, like Whitehead's simple occasions of experience. Kind of like notes, tones, and sound of an orchestra warming up, a cacophony happening everywhere. Being is everywhere, but it's meaningless and basically noise. How could they be organized, orchestrated in the brain for full, rich, conscious experience, something more like music or a symphony? Now, one more point before I get on to that is that Roger's retroactivity that he briefly mentioned in his talk on Tuesday, which erases unselected curvature and avoids the need for heat in the collapse, can also rescue free will by going backward in time. And I'll come back to that point. But basically, after reading Emperor's New Mind, it was clear that Roger needed a quantum computer, or occurred to me anyway, that Roger needed a quantum computer in the brain which could biologically orchestrate quantum information processing, to halt or terminate by objective reduction, connecting to space-time geometry, non-computable Platonic values perhaps, and qualia. And 3, regulate functional neuronal and synaptic activities, the biological end of it. So it occurred to me that microtubules were the best bet, and I suggested them to Roger, and fortunately and happily, he agreed they were good candidates. So we developed a theory, and on the left, you see the Orch OR qubit. For a quantum computer, which we eventually want to collapse by OR, you need a system that can be in 2 different states and a superposition of both states. So our first idea, my idea, was that the protein conformation changed, but it was triggered by pi resonance electron dipole oscillations in among just at least 2 of them that we see inside. Now, in 1999, the atomic structure for tubulin came out, and I remember J.Tuszyn excitedly telling me about it, and we learned a lot more, and we'll come back to that. But it caused us to change the qubit in light of what we now knew, or what we then knew about tubulin. And so from 2002 to the present, we've used this qubit shown on the right. I'll explain that in a minute. And what the evidence from the atomic resolution showed that there were 86 pi resonance rings in tubulin, 8 tryptophan, 36 tyrosine, and 42 phenylalanines. And you can see them in the 3 different colors. And the red spheres are where anesthetics bind. And this was published by Travis Craddock in 2012. These pi resonance groups are important in quantum coherence and photosynthesis to make energy and food in the nonpolar pi resonance clouds. And they are the basis of organic chemistry, which started with started with hydrocarbons, alkanes and alkenes, but chemists were faced with the molecules C6H6 that they didn't have a structure for. And Kekulé had a dream that the hydrocarbons were a chain, and in his dream, one of the chains were snakes, and one of them swallowed his tail, and became a ring, and he woke up and said, benzene is a ring, and he was correct. And benzene has 3 extra electrons, and they tend to form these delocalized electron clouds above and below the hexagonal ring. And these electron clouds support electric and magnetic dipole oscillations, excitons, charge transfer, phonons, fluorescence, quantum stuff, quantum activities, suggesting maybe that life and consciousness derive from pi resonance in microtubules. Now, these pi resonance, these molecules attract each other, and when they get close to at the van der Waals radius, they induce paired dipoles, which then oscillate in terahertz. So these are benzene rings. If you in bulk, they go in your gas tank and drive your car. But if you put them at the right spacing, they have very interesting, delicate quantum properties, including oscillating in terahertz. Also, they oil and water, oil avoids water, and the oily pi resonance group tend to coalesce inside proteins, upper right, inside lipid bilayers, lower left, and inside nucleic acids, lower right. They form an internal pi resonance, what we call a quantum underground of nonpolar organic rings pervading the core of living systems. So this is where the quantum stuff can happen, deep inside where it's protected, and there are fewer reduced degrees of freedom. Should also mention these pi resonance turns up pretty much wherever there's something interesting quantum biological going on. For example, yesterday, excuse me, we heard about this experiments by Marcus Arndt's group of putting porphyrin molecules in superposition. And I just want to point out that porphyrin molecules are largely pi resonance with a lot of pi resonance groups, and they more easily go into superposition. Also, I've gotten interested recently in astrobiology and panspermia, in which life is suggested to come from other planets, asteroids, interstellar gases, and so forth, as polycyclic aromatic hydrocarbons and clusters of amphipathic molecules with pi resonance rings and gigahertz and terahertz oscillations. And there's a astrobiologist at University of Arizona, Dante Loretta, who is in charge of a big mission bringing back samples from an asteroid, who thinks that consciousness and life are very closely related, and this may also be a clue to the origin of consciousness outside the Earth. We'll see about that. Getting back to the pi resonance groups, if they form two-dimensional sheets, they're in graphene, and when these are overlaid at specific magic angles, superconductivity and moire patterns occur at fairly warm temperatures. So there's something going on with these pi resonance groups and quantum biology. And finally, along these lines, psychoactive molecules, dopamine and serotonin, and all the psychedelics have a lot of pi resonance associated with them, and they're mentally and psychically very active. Which raises the question, which came first, consciousness or life? Neuroscience and philosophy would say that life came first and consciousness emerged from it, but panpsychists, naturalism, Eastern philosophy, and including the OR theory would say that consciousness preceded life. OR was in the universe before life appeared, and life therefore could have emerged from consciousness. And life apparently began in a primordial soup, simmering mix of biomolecules 3 to 4 billion years ago. And this was simulated in the 50s by Miller and Urey, and they added lightning and other things and found organic amphipathic molecules. This is what they found in their simulated primordial soup, dopamine-like molecules with nonpolar oil-like rings on one end and polar water-soluble tails on the other, which coalesced to form micelles, shown here, which could have occurred in the, and this is my speculation, I'm not sure Roger will go along with this, but back in the primordial soup, they could have formed these micelles and begun to cooperate and reach threshold for OR and had a very simple conscious moment, which most likely would be random, but occasionally could be pleasurable, feel good. And this could have been highly significant because this could have prompted, this could have been a feedback fitness function for life to develop in order to feel good, to optimize pleasure, which makes more sense than optimizing your genetic survival, I think. Just to speculate a bit further, pi resonance rings, pi stacks have two stable arrangements, T-shaped and parallel displaced. And maybe it's something like this, as simple as this. This is tongue-in-cheek, but not entirely. Something like this at this basic level could be giving rise to primitive feelings that have to come from somewhere, and the emergence ideas at high level don't seem to be working. So Darwin is a pillar of science, but behavior is driven by reward. There were no genes in the primordial soup, and evolutionary theory, even though Darwin didn't, but evolutionary theory ignores consciousness and feelings. So maybe feelings are driving evolution and continue to do so, not just hedonism, but altruism and spirituality and so forth. So getting back to microtubules, here we have the tubulin molecule with the pi resonance groups. And here we see on the left the aromatic amino acids, where they are in the tubulin. And on the right is a simplification that we did for our model, showing that the pi resonance groups in the tubulin can be aligned and form the kind of dipoles that I showed earlier, where they attract and oscillate in terahertz. And this led to our qubit model of a helical helical pathway of dipole oscillation in opposite directions and in both directions, following the helical winding pattern of a microtubule, in this case an A-lattice microtubule. And therefore, Bing would occur when a critical number of tubulins were in superposition at time t equals h bar over E sub g. So we calculated E sub g for tubulin in three ways: separation superposition at the levels of 1, the whole tubulin protein; 2, atomic separation at the level of atomic nuclei; and 3, separation at the level of nucleons, protons and neutrons. And separation at the level of atomic nuclei was the dominant high energy effect. And so we used that to calculate how many tubulins we would need for a given time cognitive or conscious event. We know that there's about 10 to the 8th to 10 to the 9th tubulins per neuron, 10 to the 19th to 10 to the 20th tubulins in the brain. By t equals h bar over E sub g, for 10 to the minus 1 second, for 100 milliseconds, cognition EEG, for example, you'd need only 10 to the 9th tubulin. It's a very small number, and you'd need to maintain coherence and avoid decoherence for a very, very long time. So we favor a much shorter Orch OR period of, say, 10 megahertz, 10 to the minus 7, this would require interference and to give cognitive events, but has a favorably short decoherence time. And for 10 to the minus 7 seconds, we would require 10 to the 15th tubulins. This is about a 10,000th or 100,000th of brain capacity. But this is feasible because we'd only have to avoid decoherence for a very short time. But we require interference. If we plot the number of tubulins versus for Orch OR, we can see that we can rank, this is from a paper with Allison Muatri. Allison's going to talk a little bit later. We can plot based strictly on the number of tubulins, the intensity of experience, or the level of possible consciousness, from a human maximum going all the way down to a mouse, cerebral organoids that Alysson will talk about, C. elegans, single cell, and even plants, which have a small amount of microtubules. And the frequency and the intensity of the experience would go down as you go to the left and downward. Great minds think alike. A number of people have come to the idea that quantum electron pi resonance states are important for life and consciousness. Schrodinger had this idea. Jean Georgi had this idea. The Pullmans had this idea that quantum behavior of electrons in pi resonance clouds support life and consciousness. And Herbert Froelich had this idea that coherent dipoles in nonpolar regions in geometrically constrained proteins, membrane proteins and cytoskeleton, were essential for life. Philip Stamp kind of badmouthed Froelich coherence yesterday, but I think if you look at what he said about it occurring in nonpolar regions, that's quite a good model. It's kind of a meta model, so if you get too specific, you can mess it up. But if you look at it in the nonpolar regions, it works quite well, and it's consistent with where anesthesia works. So overall, we have a kind of a multi-scale hierarchy Now, a lot of people have these hierarchies going from the individual neuron upward into networks in the brain, but we extended downward from the pyramidal cell to the individual microtubule to the rows of tubulin to the pi cloud dipoles, and then all the way down, skipping over to the Planck scale here. And here we see some oscillation frequencies that go along with this. And this is mostly garnered from the work of A.Bandyopadhyay, who will be talking a little bit later, who actually has evidence to support cell-similar patterns, triplets of triplets, he calls them, occurring at terahertz, gigahertz, megahertz, and kilohertz. And this would enable Orch OR to occur all the way down into, down into kilohertz even. And here's another view of the hierarchy. This is a little bit different version. And this extends all the way down, includes atomic nuclei and alternate space-time curvatures. So how do we test for this? Quantum spin transfer through pi electron resonance, benzene rings, and superposition were shown in 2003 by Ouyang and Aushalom, just connecting quantum dots by benzene rings, you can see on the left. And they got quantum superposition and interference that you can see on the right. And more recently, looking at the photosynthesis FMO protein, Engel's group used 2D electron spectroscopy to show these sawtooth interference beats indicating superposition of the electron cloud, pi resonance electron cloud dipoles in the protein. And we hope to do this for tubulin. And we'll be hearing more about the experimental approaches from my colleagues a little bit later. And then we'll test relevance to consciousness with anesthesia. So just to wrap up, let me say something briefly about free will. I said that consciousness occurs in the third wave, too late for conscious response, which occurs at 100 milliseconds. We often consciously within 100 milliseconds, consciousness is considered epiphenomenal and conscious control an illusion. And Roger mentioned the work of Ben Libet, shown here, who in 1979 did an amazing experiment on patients who were awake during neurosurgery. And you see this person looking who's actually, you should envision him lying down, and we're looking at the top of him on the operating table. And Libet was able to use, the brain was exposed. He could do direct cortical stimulation and recording from the brain. He could, for example, the finger area of the brain, he could stimulate the finger, and he could talk to the patient and find out when the patient felt an experience. So what he found was that both an evoked potential at 30 milliseconds immediately after stimulation and 500 milliseconds of brain activity are required for near immediate conscious experience. And this is a paradox. Let me...
we're running low on time, Stu, if you could wrap up.
Okay, I'm wrapping up. This shows that the evoked potential occurs, and on the left, you need cortical stimulation for 500 milliseconds. If you give thalamic stimulation, there's no ongoing activity. Therefore, Libet concluded that there had to be backward time effects from the time of neuronal adequacy to conscious experience. This can be explained by Rogers' retroactivity, which erases unselected curvatures. And this is fleshed out in this paper here. In conclusion, let me say that quantum effects occur within biomolecules in a nonpolar quantum underground defined by where anesthetics bind and act. The Penrose Hameroff Orch OR theory is based on orchestrated quantum computations in microtubules inside neurons, which terminate by Penrose objective reduction, connecting the brain to the fine-scale structure of the universe. Orch OR has more explanatory power than other theories, is falsifiable, and currently being subjected to critical testing.
All right, excellent talk, Stu. Okay, so next up we have Dr. Greg Scholes, who is the chair of the Department of Chemistry at Princeton University and the director of the BioLec Energy Frontier Research Center. He's the editor-in-chief of the Journal of Physical Chemistry Letters and co-director of the Canadian Institute for Advanced Research Program, looking at bio-inspired solar energy. Dr. Scholes is a pioneer in the emerging field of quantum biology, where he has worked to demonstrate experimentally that quantum coherence can be achieved at room temperature within biological systems, and that coherence is not only possible in the warm, wet, and noisy environment of our bodies, but coherence is essential for biological function. He has published frequently in the most prestigious journals that one can publish in, demonstrating his academic excellence and his ability to push the sometimes reluctant field of biology into a new paradigm. So let's welcome Dr. Scholes to the stage.
Thanks very much, Justin, for that kind introduction, which I didn't write. Okay, I'm going to carry on in a similar spirit to Stuart. It's going to be an overview of some thoughts, a little bit of results, mostly some thoughts on quantum phenomena in the brain. What should we be thinking about? And this is inspired, again, by Roger's books, in particular his more recent one, Shadows of the Mind, where he raises questions. And as he said in his introductory lecture, he wrote these books thinking about how does the brain work, and as Stuart emphasized, perhaps more deeply, how does the mind work and what is consciousness? And so I want to try to make some connections here, or ask some questions about where connections might lie between these questions and science that we know, pretty broadly speaking. Let me move this. Brain, what we do know is that brain function, just how it works mechanically, is facilitated by a hierarchy of networks. And I'll talk a lot about networks, and these are networks at many different scales. And that's well established. And the point of this is, this is an incredibly complex system, and this is going to become relevant when I ask questions about quantum mechanics. So Stuart, for instance, and I quote him here, in a paper where he's talking about how anesthetics work, says, this is not a question that's easily answerable because the mechanism by which, and I can't read this, here we go. The mechanism by which brain physiology, oh my goodness, sorry, I keep blocking my text. Brain physiology produces consciousness is unexplained. And there are experiments, I show some here from this particular paper, studying the way networks are connected when a rat in the brain, when a rat is awake versus under the influence of anesthetic. There's large length scale changes in these networks, and they talk about the loss of cortical integration. So part of the point of this is that if we're going to address questions about how the mind works, how the brain works, and of course this is well appreciated, we can't think at the level of the microscopic scale that I'm used to thinking about as a physical chemist. And this is, of course, very relevant when we start thinking about quantum effects. And Roger pointed this out explicitly in his book. He said we must think beyond a single protein or a cell. Relevant effects span vast numbers of cells. So he goes on to say that we must seek quantum systems that retain their quantum nature on incredible scales of size and complexity. And this is the key challenge, obviously. So it's very easy to very quickly dismiss this as impossible. And this has been done. So what one can point out is that based on knowledge that we have and systems that we have studied, that we can set up quantum coherence in, those quantum effects, the entanglement or the coherence or whatever the non-classical correlation is lost very quickly through decoherence. And so I figure I can just explain what that concept is really quickly as dephasing, actually, that I'll show in the picture. And we can just imagine a pendulum here to indicate the the quantum system. So quantum systems need to have some phase information that's in the wave function. And we can imagine that as a harmonic oscillator. If we have a single pendulum with a single length rope, it's swinging backwards and forwards, it will swing at a particular frequency. If we imagine a room full of these pendula with slightly different length cords connecting them to the ceiling, each one will have a slightly different frequency. So even if we start them all together, release all the balls and watch them swing, initially, as you see in the simulation on the right, they'll be swinging in phase, but because they each have slightly different frequencies, they'll get out of phase. And when we make an ensemble observation, we'll say, well, there's no coherence in this room. And that's the process of dephasing. Decoherence is a similar process, but it would be a dynamic change in the length of the cord of a single pendulum. And Philip Stamp has a nice recent paper on discussing the difference between dephasing and coherence that I just saw recently. So coming to quantum superpositions, can we study them? Can we produce them? Yes, and I'll show you that briefly with some experimental results to illustrate, before I start the main body of my talk, why it's easy to say quantum effects are impossible in complex systems. So we can make a superposition, for instance, of excited states of a molecule or atom, shown in the diagram on the left, taken from an old paper, where if we had a high frequency excitation light, as you know, as you tune the frequency, the energy gap between the ground state G and these excited states, we could tune the frequency and be resonant with A. We would know we would excite state A. We could then increase the frequency of our excitation light, tune it to the blue, and we would excite state B. Now, with a short laser pulse, because it's short in time, carries a broad bandwidth of frequencies. When we excite a system like this, typically what we do is excite both states together synchronously, and we don't know which state, A or B, we excited because the bandwidth of the laser covers both. It's almost a white light. And when this is the case, the system evolves differently. So there is the possibility we excited state A and the possibility that we excited state B. But in addition, there's the possibility that we excited them both in coherence. And we see this typically by an interference, which in the equation here, it's that cosine term. So it's an oscillation that occurs with frequency of the frequency difference between states A and B. So we can do this experiment in different ways with short laser pulses. This is an example with an experiment called 2D electronic spectroscopy, which makes a map, which is the middle picture on the top here, a movie, in fact, of these maps, which shows a correlation on the y-axis between the excitation energy and on the x-axis between the detection energy. These are difficult experiments because we need to resolve the phase of the radiated signal from the sample after the nonlinear response to the excitation sequence. And so here's the example on the bottom middle. These are colloids, they're semiconductor nanoplatelets. And the reason we've studied them is that they're atomically precisely thick. Okay, the actual shape, when you look down at the large shapes in the electron micrograph, there's dispersion there, but in the thickness, which you can see on some of the plates that are aligned right, is very uniform. And so that's reflected in the absorption spectrum. On the top left, you can see the two electronic states, the two exciton states of the nanocrystal, the HX, LX, heavy hole, light hole excitons, are well separated. And the red is the laser pulse, which, as I said, is broadband. It excites both together. So we make that superposition. And indeed, when you go to the top right there, when you look at cross peak in our spectrum, where we've created the superposition with the excitation, you see these oscillations at the frequency difference between these two electronic states. There's electronic coherence. However, what you see is that it doesn't last very long. And this is what people point out when they argue against quantum effects. So this dephasing time is 13 femtoseconds at room temperature. These systems, and this is in fact most likely dominated by dephasing, not decoherence. So this, the oscillators, each nanoplatelet having some roughness or heterogeneity has slightly different frequencies, and they run out of phase really quickly there. So if we sum up the top here, the amplitudes of all our oscillators, we sum those amplitudes and just look at the ensemble average here on the bottom, you'll see these oscillators appear to, it looks like a damped oscillator. That's what the dephasing is that we measure. Similarly, we could do it in more complicated systems. We can isolate proteins from photosynthetic organisms like these algae. You see that beautiful bright color. Proteins that specialize in harvesting sunlight. It's the first step in the solar energy conversion. And in these particular algae, these light harvesting proteins with high resolution structure shown on the left here, they bind these 8 different colored chromophores whose absorption, because they're chemically different, spans a range of wavelengths you see in the spectrum here. And I've positioned where they absorb with the colored sticks, where their center wavelengths are. The key ones that we're interested in are the green ones in the center and the red ones that are on the outside of the protein. You see they go from the green to the red wavelengths here. So the 2D spectroscopy, when we excite this protein across all of those wavelengths, is at 77 Kelvin. Actually, you see here, and you see in the movie, as I said, these 2D spectra in a movie, you see these oscillations in the cross peak there. And that's very high frequency. That's 1,600 wave numbers. That's the frequency difference between the red chromophores, which have the peak here on the diagonal that you saw in the absorption, and the green ones way up here. This was also, that's lasted hundreds of femtoseconds. It's the same at room temperature. It's just that the resolution isn't so clear in the experiment, much longer than the nanoplatelets. The reason is that this electronic coherence that's underlying this, the different colored chromophores in superposition, is being driven by resonant vibrations of the molecules. So these vibrations can help sustain that coherence. That's called vibronic coherence. Okay, so what about the brain? The systems that I just showed you, even the photosynthetic protein, are incredibly simple, almost toy systems in comparison with the brain. So is it possible, there's a number of questions here that are interesting, I'll highlight two. Can quantum effects yield function from systems so large and complex as the brain? And first question is how? Because I've already said, many people would say that's impossible because of what I just showed you with dephasing and decoherence. Second question is, which I won't address here, it's a rather deep question, I think. What is the quantum advantage of that function? So I turn to further quotes from Roger's book here. You know, he makes some interesting points here. Why limit ourselves solely to what we know? He says the ingenuity that this should say that is to be found in biology should never be underestimated, which is true. You know, after 3 billion years of evolution, it's incredible what you can discover. In particular, the function of these photosynthetic systems that I know well. So this sentence that he says, why limit ourselves solely to what we know? I think the question is, or how I might, what I'm going to convince you is that there's merit in asking the question. We understand that quantum effects are fragile on these small scales in small, well-defined systems. I think that's beyond doubt, except in very special cases in environment. But is it possible that at a certain threshold of complexity, quantum effects represent a stable phase of the system? That's completely speculative, but I think it's an interesting discussion point. We're familiar with synchronization. Here's examples from autonomous systems. So flocks of starlings, pedestrians, these schools of golden stingrays are pretty examples that we can show, and we can understand how they remain synchronized in these shapes with their movement, and they can be studied in detail. So the march of locusts, I think these particular ones in this study were from Africa, have been studied, and it's shown that at a particular threshold for the number of locusts marching together, they all of a sudden coalesce into a uniform army migrating together, and this is shown from the paper that I quote here from their data and their simulation. So this critical threshold is an important in many of these synchronization phenomena. A few locusts will not synchronize. Hundreds of thousands of locusts will synchronize. There's many examples of this. It's reminiscent, these kinds of thresholds or sudden transitions are well known. And I'm not going to give hundreds of examples, but I particularly like the example of the famous Erdos-Renyi random graphs, which you can show without numerical calculations, in fact, have a very sharp threshold for the onset of what's called, or what was originally called, a giant graph, where, how to think about it is this. On the left, what we've done is just thrown a bunch of points on the ground. These are called vertices of the graph. And then there's some probability, it's shown under each graph, that we can draw a connection between any pair of points on the graph here. And so the probability is very low. Really all you see is the vertices with a couple of connected points, which then get put in the middle of my picture. So we can see them as the probability increases. Again, no surprise, you get a few connected bits and a lot of scattered vertices, but at a critical probability that's related to the number of vertices here, you end up getting a dominating single fully connected, or not fully connected, what I mean is a single graph that there's a connection running through it, through all the vertices throughout the entire graph. And this is a phase transition. So it's amazing. At a particular threshold, all of a sudden you go from a random scattering of vertices, you might think of these as neurons if you like, to a network that emerges out of nowhere. Other types of synchronization that are interesting can be mechanical in nature. I like this paper by Strogaerts that examined a phenomena in the Millennium Bridge in London, where it turned out when there was too many people walking across it, and as you saw in the earlier picture, pedestrians in a crowd synchronize in lockstep, that was enough to couple to the modes of the bridge and cause it to sway. So again, it's an emergent phenomena. You don't see that until there's a threshold of number of people walking on the bridge. We're used to, or we're, very commonly also in biology think of synchronization as a phase oscillators. Typically the cells in the heart, they don't fire randomly, obviously, but they fire in sync. And this is ensured by small interactions between the cells that provides feedback. These are nonlinear oscillators, which are well studied. I first studied a few hundred years ago, I can't remember when, by Huygens, who was laying in bed, so the story goes, not feeling at his peak, and he was watching two pendular clocks on the mantelpiece and noticing that the pendula were in sync. And he found that no matter how he started the pendula, no matter what initial conditions he gave them, they would ultimately synchronize. And then again, these are coupled oscillators. They're weakly coupled, this one through the wood. You can see at the top here, here's by the shelf, of course. So weak coupling between oscillators can cause them to synchronize. Synchronized fireflies that you find in certain national parks here in the United States are a classic example of this, and it's modeled by very elegant and simple theory called the Kuramoto Model, which is well known. And here's the point that I've been evolving to here. If you remember the decoherence picture that I showed you, the downfall of quantum effects in complex systems, we'll look at what happens in these coupled oscillator systems. We can start, in fact, this is just a few oscillators out of hundreds. In my simulation, we start them out of phase. They become in phase. They synchronize with time. They become in lockstep. It's absolutely opposite of dephasing. These are classical oscillators. But imagine if we could leverage this for quantum mechanical oscillators. What it would mean is that while a few building blocks of our system may be unstable like we're used to in the quantum world, but 1 million of them may cross the threshold and be a stable quantum phase. One can bridge the gap here between these classical oscillators and start to study quantum mechanical properties. One can start by looking at spectral properties of graphs, for instance, which I'm not going to go into what this is all about, except to say that, you know, these are the circles in each case of the vertices, and the solid lines obviously how they're connected. In quantum mechanical models, the connection is the coupling, the weak coupling. So these Kuramoto oscillators, which can be have different architectures, one can predict the properties of these. And I wanted to do that to point out that if we study these different kinds of networks, we can ask the question, well, is there, how to phrase this generally, is there the possibility that they can sustain quantum mechanical correlations even in the face of noise? Okay, how stable are they in noise? And the metric I've used in this particular graph is von Neumann entropy. When it's low, we're close to a pure quantum mechanical state. And certain architectures of these graphs are much more stable in the face of noise than other architectures for the same size network, and all other parameters the same. Interestingly, the star graph, this one here, so one oscillator coupled to all the others, can be realized experimentally in what's called cavity QED, when we sandwich molecules, high concentration of molecules or atoms inside between two mirrors, tuned, so the cavity is tuned to the resonance of the molecules. And what you find is one eigenstate split off to low frequency called the lower polariton state, and 10 to the 5 states typically are left here. They're called dark states. But this single quantum state is this super stable state here with very low entropy. To put this just in perspective here, this is this is not a 10 to the -5 wave number effect. This is huge. Here's the absorption spectrum in red of a film of these molecules that we've studied. And when we put them between the two mirrors, the absorption spectrum, the cavity was tuned to where that blue bar is. The absorption is shifted here, hundreds of nanometers, to this nice sharp peak. That's the light matter hybrid state. But that's very low quantum mechanical entropy state. In other words, it's a very robust oscillator that incorporates hundreds of thousands of molecules. Okay, this is very different than the fragile few molecule quantum states that I showed the experiments of earlier. It's interesting. This is an emergent phenomena as well, as these are simulations again, but as we tune the cavity strength, how strongly each molecule couples to the cavity, and each molecule couples very weakly to the cavity. This is the same coupling that gives rise to fluorescence. Very, very weak for each molecule. But as we improve the properties of the cavity, all of a sudden there's quite a steep onset, and we go from no molecules coupling to the cavity to thousands in the simulation. And that is correlated with this onset of coherence. And you can calculate, this is actually relative coherence. This is the coherence of that ensemble relative to the nearest mixed state. So this is the real onset of coherence of that system. Again, it's an emergent phenomena. Each interaction, as I said, is ridiculously small, less than a wave number. But the collective effect of 100,000 molecules in the cavity, it produces this incredibly robust state. Okay, aside from ignoring what happens with population, loss by fluorescence and so on, trivial loss of the population. So I deliberately made this talk not ending at the threshold of my time, because I wanted to make this fairly key point here, I think, that I've summarized here. At small scales, coherence is fragile, and that's where it's easy to say, well, quantum effects are impossible as the system gets more complex. But as I said, as Roger pointed out in his book, why be limited by what we know? So if we think about how coherence, classical coherence, emerges as a stable phase when you have a threshold number of locusts, starlings, pedestrians, whatever it is there the possibility that a quantum phase can emerge when the system reaches sufficient complexity? And this could provide the answer or a clue as to how these networks in the brain maybe are more than what they appear. So thanks, Stuart, for organizing this symposium. Thanks, Roger, for the inspiration, and congratulations on the Nobel Prize, and happy birthday, and thanks to my group. So I figured I'd leave some time if we're allowed to have discussion. So thanks.
Yeah, thank you, Greg. Yeah, we have some time for questions, 13 minutes or so. So yeah, if people want to put questions for Greg in specific in the chat, please do so. One question for you by Robert Bernstein. If what you say is true, can you design a quantum computer that would work at room temperature?
Right. Well, that would make sense. I couldn't, but yeah, that would be the idea. Would be that yeah, sure. Yeah, maybe we can't make a quantum computer with 10 bits, but maybe it's easier to make one with 1,000,000 bits. That would be the speculation. It's complete speculation. Although I didn't go into. There is science behind a lot of this, which I didn't go into the details.
Okay, I'm looking for a question specifically for Dr. Scholes. One question I have, yeah, so you're talking about this like emergent coherence that can come about. Would this be like the equivalent of quantum coherence that people talk about, or could this be happening at the level of like electric fields and neural oscillations? Do you sort of draw a distinction between what's possible at that microscopic level, or is it like a distinct phenomena, or is it just coherence more generally?
Yeah, more generally, typically, or oftentimes these can be scale-free phenomena. So there's coherence, at the microscopic level and the macroscopic. Philip Stamp had a question apparently, but he might be able to say it with words. Philip, can you unmute?
Yeah, I can't get this question and answer thing to work on my computer, but I can apparently type in a chat. So my question is this: they kind of self-organize criticality, but at a quantum level that you're talking about, is, as you point out, quite common. In fact, a very old example is BCS pairing, which forces a state to drop down below the others, which is right. But the problem with all of these states that you're talking about is that they have very low entanglement. And so the question, I guess, to you and to Roger, I suppose, is, are you envisaging, when you talk about the quantum properties, quantum mechanical properties being involved in brain function, are you talking about high entangled states, or are you just talking about a simple Bose condensate?
Yeah, that's a good question, Philip. And that was, that was a question that related to a question that I put on one of the earlier slides, but declined to discuss, or haven't had sufficient time to discuss. I think this is a key thing. If it relates to this, if quantum effects are important, what is their function? What is the quantum advantage? And is it entanglement, or is it a different kind of quantum correlation? So, right, I think this is a really interesting point.
I mean, the speculation you're making here seems to me to be quite amazing, actually, if you put it in the following way. Up till now, everybody's assumed that high entanglement states are very fragile. But what you're basically suggesting is that maybe there's some kind of high entanglement state that is not. But now, the states that you've been mentioning don't have high entanglement, but to paraphrase Roger, why shouldn't we speculate about that? I don't know of any known mechanism for doing that, but
Right. Yeah, I think it's worthy of study. Yeah, exactly. These in the ultra strong coupling regime, you can start to get higher correlations.
That's right.
Higher entanglement in the polariton states.
Yeah, no, that's exactly what I thought you were saying. Yes. But,
yeah.
All right. Well,
Roger has his hand up. Justin.
Oh, Roger, go for it. Yeah.
Can I? I think that we need a little clarification in the terminology. When people talk about quantum effects, I always find this very confusing, because the whole of chemistry is quantum effect. When you talk about quantum effects, you're obviously not meaning chemistry, because that's accepted as being something which is the primary ingredient of all these things. So we mean non-chemical quantum effects. I don't even quite know what the terminology means here, but it certainly means non-chemical. There's a certain other question of terminology which confuses me too, well, two other questions of terminology. One of them, the effects that you've been talking about, that's Greg. These things depend on non-linearities. I think at that point you weren't stressing. I mean, the fact that what you're looking at are non-linear effects. So if you just look at the Schrodinger equation, if you like, you don't get this thing. It has to be something which is non-linear. So either you're looking at a modification of quantum mechanics, which is non-linear, or something else. And I'm a little confused by that point. The third issue, we need some clarification in the terminology, is the quantum computer notion, because this is certainly not part of Orch OR. In Orch OR, we aren't thinking of the brain as being a quantum computer. It's something beyond that. Because if you're involving the collapse of the wave function, that's not part of quantum computation. Or at least the way you think of quantum computation, if you like, it comes out at the end when you actually look at the state or something. But the mechanism of the system is straight quantum mechanics. And this has to be beyond quantum computer. So I just thought I'd make these points, because I think the issues weren't quite clarified to the extent I'd like.
Right. Yeah, I absolutely agree, Roger. I can quickly address them. Yeah, your last point probably doesn't need much further comment. What I'm thinking is, I'm thinking more abstractly about the properties of the network, not as it being a quantum computer and so on. In fact, it would probably be a network that influences another network, like Jeff Hinton had discussed in the past with learning algorithms. But your comment on the nonlinear effects, which is the Kuramoto model, coupled oscillator model, is really interesting. Yeah, absolutely. Quantum mechanics is linear. It'd cause a lot of problems if it was completely nonlinear. I mean, as you're aware, there's work of thinking about small nonlinearities that might explain collapse of the wave function and so on. But forgetting about that, what I do wonder with this comment is it possible, the other side of the coin is it possible that nature started this very simple system? It started with quantum mechanics, but evolved away from that because these nonlinear systems are so robust. Does nature prefer to use the nonlinearities of classical systems in order to leverage the robustness? That would be the other thought that I have, in which case it would take us away from the idea of quantum effects. And your comment at the start of the trivial, where are we drawing the line between the obvious quantum effects, a molecule is quantum, and this other stuff? Yeah, that's a good question. We usually call these non-trivial quantum effects, which just is maybe legal terminology, I'm not sure, but yeah, these would be quantum effects on length or time scales that are not obviously important for the physical system.
Yeah, a couple more minutes here. Yeah, to kind of riff off of that a little bit, there's some questions in the chat. You mentioned the scale-free idea of these emergent coherence patterns. Is there a limitation on the higher end? If you have social interactions or interpersonal communication, yeah, what's to say your principles don't apply at that level?
Oh, right. I say, oh, wow. Yeah, mind reading and all of that. I think, how to think about that is that you need to factor in the strength of the interactions between nodes in the network. So, this is what these small world, speaking of social interactions, these small world networks that are known in sociology, so 6 degrees of separation, etc. It's all about the connection. So it's not the fact that you have the entire population of the United States, with connections among it. It's that there's specific clusters of connections within that. Okay. So scale-free doesn't mean a fully connected graph. What it means is that at any scale, whatever the x-axis is that you're looking at, you see similar trends.
Or holographic interference pattern from a low level could give you what you're looking for. Something like the holographic model back in the 70s?
Right. Yeah, I didn't. Yeah. I mean, I didn't nail down specific, physical instances. I was just thinking more generally on any kind of network. So whether it's the neural network, whether it's the microtubule networks, any kind of network that where we might want to, where the network can be huge and we can leverage the size of it. That is the key. So it's a synchronization. So in other words, so in your talk, Stuart, you discussed, of course, that neurons fire You know that there is a synchronization in the firing. I forget what it is, 5 hertz or something. And so it would be.
Well, that's complicated because EEG synchrony comes from the dendrites and soma before firing, and firing may or may not be synchronized. A lot of people confuse that and think firing is what's synchronized, but in EEG, for example, gamma synchrony, that's all postsynaptic dendritic somatic potentials.
I see. Yeah.
I was a little bit worried, I guess, when you were talking about, do you think complexity could lead to quantum this high-level quantum effects? Because I've heard, complexity kind of thrown at problems too often. I mean, it'd be great if it worked, but I think something more like a hologram would be better, or some kind of geometry.
It's not complexity per se, because of course, if there's disorder is going to beat everything, but it's leveraging numbers. So there's emergent phenomena typically, if well, it depends on the particular structure of the network, what it is, but it's a network with particular kinds of interactions, and it can be that at a particular scale, synchronization in the network beats the dephasing. Okay, and I didn't show calculations of this, but I showed some simple examples. But that's right. So the idea is that at a threshold, so how to think about it is that with a small system, I showed those experiments where the decoherence, just destroyed superposition states, 13 femtoseconds, for example. And so what we might, so not in that particular case, I would say, but we might imagine, let's say that is the case on that scale, but let's say we can have a system that's networked that at a certain scale, the emergent properties of that network defeat everything else. So the most simple way to think about it is that in the spectrum of the eigenvalues of the system, it spits out a lower eigenvalue that lays below most of the eigenvalues. That's how you're going to beat energy disorder. That's what happens in that star network. So there's a clear reason why this is the case.
All right, I'm gonna have to stop it right there as the session chair. But yeah, thank you, Dr. Scholes, for your amazing, interesting, compelling talk. All righty. Next up, our third and final long form talk before we get into the panel will be Dr. Allison Muatri, who is a professor at the Department of Pediatrics and Cellular and Molecular Medicine at UC San Diego. He is the director of the Stem Cell Program and the director of the Archaeolization Center. Allison views the brain through an evolutionary lens, seeking to understand how genetics, lineage, and social pressures shaped our development. Recently, Alysson has been working with brain organoids, stem cell-derived cooperative assemblies of roughly 2.5 million cortical neurons in order to study the emergence of complex neural activity patterns. Recently, Alysson has teamed up with Stuart Hameroff with the prospect of using a cerebral organoid to test the orchestrated objective reduction theory of consciousness. I'm looking forward to see what he has to talk to us today. So let's welcome Dr. Allison Watry to the stage.
Thank you. Let me see if I can start sharing my slides here. Just a moment. Can you all see these slides? Is all good?
Yep.
Yep. All right. Fantastic. Okay. So first, I would like to thank Stuart for the invitation. Congratulate Roger for the achievements and birthday. It's my honor to be here and share a little bit of what we do with you guys. So I'll focus first on a brief introduction of the brain organoids. I'll guide you through the most recent developments that we've been done. And then I'll dive into some of the potential experiments that Stuart and I have been cooking with these organoids. So we mostly use brain organoids as a proxy for the human brain, especially because lots of neurological conditions that we study here in the lab, such as autism, epilepsy, they all have embryonic oranges. And the human brain is very inaccessible experimentally in utero. So we have to rely on tools that don't have the right resolution that we want to really dive deep into the synapses or neurons and studying how the networks are happening or forming during very early stages in the brain. So if we don't fully understand that, of course, we don't understand how the brain works. So we see that that would be like a really nice tool to understand the early stages of brain development, how we can achieve more complex networks and perhaps bridge with human cognition. It's totally in vitro system, but I think it's a first step towards that. And so these two brain organoids are three-dimensional clusters of cells that are derived from human pluripotent stem cells. These pluripotent stem cells can be derived from individuals that we can reprogram, so they capture the genome of the individual, or could be derived from human embryonic stem cells from the blastocyst. These usually come from in vitro fertilization clinics that dispose the unused embryos for science. And the first person who actually showed that these cells can neuralize and form this three-dimensional structure was Yoshiki Sasai from Japan. This was in 2008. I would call that the first brain organoid paper. He showed that the cells will self-assemble and form structures that anatomically look very similar to the human fetal brain. And here are some of the images from his pioneer work. These protocols have been evolved over the years, and now we have a variety of differences and modifications that can create different types of brain organoids. Basically, we divide them in two types. One is called unguided differentiation, where you can see multiple brain regions in the same unit. So sometimes you have a piece of a hippocampus, a cortex, together with a thalamus, not necessarily in an organized fashion. So they are all disorganized, most likely like teratoma, but the pieces of the different parts of the brains can be seen there. And there is advantage on or disadvantage depending on the questions that you have. The other type of protocol, and this was the one initially derived from the Sasai lab, is when you guide the cells, the pluripotent stem cells that are quite naive at that stage, but by adding factors to the cultures, you instruct the cells to acquire certain brain identities. For example, I can derive a cortical organoid, or I can derive a thalamic organoid, or a retina, by changing the factors and the combination of factors that I can add into the tissue culture. So I can drive the cells to those different things. You can also combine these organoids, and they normally fuse to study different brain regions or the interaction between different brain regions. So these are most of just a big summary of the different protocols that are out there. But as I mentioned in the beginning, this is an in vitro system. So there's lots of intrinsic limitations of the model. And sometimes, even scientists portray the information that these are mini brains, and the image that you get is that you have a completely brain in a dish in the lab. And that's not true. I'll show you how they look like. They don't look like this. Some of the limitations that we have because of this technology is the maturity of neurons. They're quite mature. They start from embryonic stem cells. So the nature of these neurons are quite immature. Because they're not vascularized, they cannot grow past 0.5 centimeter. So they are tiny. You can actually see a naked eye, but they don't grow any bigger. There are recent efforts on vascularize these organoids so we can add more neurons, but we're not quite there yet. And because of that, we don't have all the cell types represented. So it's a simplistic view of the brain. And even, the type of culture condition that we grow them, we are not 100% sure that we mimic the embryonic environment in uterus. So these are all pitfalls of the technology. And I think that between 5 and 10 years from now, we're going to solve most of those issues, and we're going to have like a much better model. But for now, that's what we have. My lab has been optimizing the media that we grow these organoids so the neurons can mature faster. We use the design of experiment. This is a century-old mathematical and statistical tool where you create matrices to understand the interplay between variables that we add in the culture. For example, all these factors that we use to drive the cells towards a specific brain region, these are all normally empirically defined based on mouse embryology, because we don't know how the human brain works. So we don't know if this is the right way to create the brain from scratch or not. So we use this tool to kind of guide us on optimization of the entire procedure. So we did that for brain cortical organoids more recently. It's actually a work that took like 4 years. But finally, we have like a very nice recipe to create a functional cortical organoid. So those neurons now start to fire. They can have spontaneous activity, something that in the past we couldn't really see in a robust way inside these organoids. So we start from single cells. We neuralize them by adding 2 factors as dual SMAD inhibitors that drives the pluripotent stem cells into cortical progenitor cells. Then we stimulate the cortical progenitor cells to proliferate by adding FGF and EGF. These are growth factors. And then we remove the growth factors and let the cells acquire their own identity. So they will specialize in different subtypes of neurons, for example. And I'll tell you in a moment what kind of cell types we have in there. So we start from single cells and we end up with 2.5 million neurons inside these organoids, plus all the glia cells and other non-neuronal cells. So this is a postdoc in the lab, Isabel. She's holding a plate with lots of these brain organoids. You can actually see these white dots. Each one of them is one of these organoids with 2.5 million neurons in there. So we can generate a bunch of them. Actually, for every single batch, we have about 1,000 organoids that can be generated, and we use for multiple experiments. So the way they self-aggregate is by following exactly how the brain does. This is all genetically programmed. What you see here is a 2-dimensional representation of one of these organoids growing. So the top is the center of the organoid. You can see some of the neuroprogenitor cells are migrate from inside out. As they migrate, they follow the directions of these cables. These are the radioglia cells that are there to guide the cortical formation. And as they reach the periphery, they stop and start differentiating and making contact or connections with the nearby neurons. So they keep doing that, generating layers and layers of cells. These are the cortical layers. So if you cross-section one of these organoids, you actually have the ventricular zone here, surrounded by these neuroprogenitor cells. Some of these are similar to choroid plexus cells. And they will eventually migrate out to form the cortical plate. Here, the radioglia cells in white, and a nice cortical plate is formed. As you age these organoids, you can keep them alive for several years. As you age them, the diversity of the cortex will increase over time. One thing that we do to characterize the cell types is to dissociate one of these organoids and to sequence the genetic information that's in there. So these are the gene expression, global gene expression at single cell level. So every dot that you have here is one of these cells that were captured, and we clustered them based on the combination of several markers that will allow them or allowed us to define what kind of cell types they are. So you can see that this is a snapshot at 4 months. Most of the cells are deriving from these neuroprogenitor cells. There are intermediated progenitor cells. The cells that we have here are mostly neurons. These are excitatory glutamatergic neurons. At 4 months, you start to see almost 2 populations in these glutamatergic neurons. The upper layer of the cortex and the lower layer of the cortex, mostly composed by layer 5 and 6 neurons. At this stage, we also see some of the glia cells. some of the progenitor cells can actually derive into astrocytes. So the astrocytes starts as a very discrete population, but over time increases and become 50% of the organoid. So 1-to-1 astrocyte to one neuron. And these astrocytes, they peak when they are about 4 to 6 months, and they help to shape the synaptogenesis. That's when we see a peak of electrical activity that I'll show you in a moment. And also at that stage, we start to see like a very discrete population of GABAergic inhibitory neurons. They also come from a pool of progenitor cells, and they will grow up to 15% of the organoid. So the organoid will have a balance of, let's say, 80% excitation and 20% inhibition. So we have all the components of the cortex represented in these organoids. But how similar are these organoids to the human brain, even with their limitations? So one way to do that is to compare the global gene expression of these organoids. And we do that using the brain span, which is a kind of an atlas for gene expression during human development. So we have this data freely available, and we use tools such as context that allow us to compare the organoids with the brain span. And this is what we see into the pluripotent stem cells. The cells look like the embryonic cells, which is what we expect. Young organoids, they are approaching, they're no longer embryonic, but they're approaching early fetal or middle fetal stages. And if you mature longer, these organoids, they will move from this early fetal to late fetal stage, and the gene expression even matches something compared to postnatally brains or 6-month-old human brain. So they are definitely aging as you keep them in culture, and more or less follow the same trajectory as you would expect for the activation of certain genes, which is good news. So somehow they are genetically programmed to do that. So, but one thing that we need on these organoids to be able to study the diseases that we want is to allow them to develop these neural networks. And I mentioned in the past that this was one of the limitations. These neurons were quite immature. So we optimized the medium, and we wanted to test if now these organoids can actually generate complex networks. We first look into individual neurons just to make sure that those neurons are active. And there's lots of tools to do that. I mean, this is voltage-dependent dyes. You just add to the cultures, and you can see the neurons firing. The neurons, we can also use like calcium imaging. So these are all technologies that allow us to visualize neurons. But we also do patch clamp. So this is a single pipette that touches one of those neurons, and you can record over time, and we see an increase in excitability, again, suggesting that they are maturing over time. But all these technologies allow us to see individual neurons and does not give us the network that we wanted. And the reason why we want to see a network is to be able to understand what's going on at the physiological level into the organoid in a more large scale. So these are two examples of coordinated activity, such as the OLA in the stadium, or the spontaneous activity when people are quite silent, but as soon as there is a go, everybody starts to cheer up. So if you are just looking into a single individual here, you might not understand what's happening in the entire stadium. So the same thing is true when you look for neurons, individual neurons inside the brain organoid. You want to see how these large-scale neural networks are happening so we can understand the evolution of these networks. So we turn into a technology that's called Multi-Electrode Array. And these are electrodes that are plated in the bottom of a dish. So you put the organoid on top, they remain with the three-dimensional structure, so you don't need to disrupt the organoids. And these electrodes will capture the activity of the neurons that are passing by over time. So you generate an activity map or a raster plot where each channel can be represented individually. So you have the information of each electrode over time. So that gives us the potential to understand how these complex networks will eventually emerge from these organoids. And that's what we decided to do. First, let me show you, why we were surprised with the data. In the beginning, we could never pass this 5 Hertz threshold because we think that most of that was related to 2D cultures that will eventually peel off from the organoid. So we cannot keep them for longer periods of time. And we want something that is close to 20 Hertz. That would be the equivalent of a primate brain. And so people have tried to do that with organoids that were not optimized before, and even at 32 weeks old. So no matter if you keep that much longer, the organoids will never cross this 5 Hertz. But fortunately, with the optimized protocol, we were able to see that starting at 25 weeks, exactly when the astrocytes start to appear in the organoid, we see an increase in, or even a peak of electrical activity increasing over time, almost exponentially, reaching these 20 Hertz when they are about 40 weeks or 9 months. So we were quite amused by this data. And if you translate in numbers, we were moving from 3,000 spikes per second to something that reached 300,000 spikes per second in the course of 10 months. So this was unprecedented in tissue culture. We were never expected to reach that level before, but nonetheless, whatever these organoids are doing, whatever they are organizing, I think it's giving the opportunity for the neurons to start to communicate in 3 dimensions that we never seen before. So that was good news. The other good news is that both excitatory neurons and inhibitory neurons are behaving like excitatory and inhibitory neurons. And we can check on that by doing pharmacology on these organoids. For example, by blocking the GABA receptors with bicurculin, you can see an increase in activity or the number of bursts, and you can block the glutamatergic receptors, and you see an abolishment of the electrical activity. So this is all good news, suggesting that both GABA and glutamatergic neurons are working as they are supposed to do. And this is not surprise, because if you do a mass spec in the media, in the conditioned media released from these organoids, you can actually see the evidence of the production of some of these neurotransmitters. More amazingly, the levels of these neurotransmitters match what has been previously described for the prenatal human cortex. So they are producing the same level of neurotransmitters as they would be expected in a more physiological network. So this is all great news for us. One thing though that we were curious about, it is, and this happened when I was presenting this data. A friend of mine, Brad Wojtek from Cognitive Neurosciences, he said, well, if you have that much activity, maybe you should be able to detect these neural oscillations similar to what you see by EEG. And so we thought that, well, I mean, if you can see these neural oscillations, that would be a very nice way to bridge the gap from the most fundamental basic science to the cognitive neurosciences. So right now, cognitive neurosciences has been linking or correlating these neural oscillations, different frequencies of neural oscillations, with human behavior. But we, on the organoid and stem cell field, we are more on the biology. We're looking at the synapses, at the circuitries. So there was no way to cross that bridge. And maybe the organoids would be one tool that we can use to start doing that. So we're quite excited with this possibility. So Brad helped us with the analysis. Basically, we took all the data from the multi-electrode arrays. We created the population spikes in the LFP. So this was the basis for the neural oscillations. And we start to see oscillations when they are about 2 months. By 4 months, we see that they become quite complex, but it's still very regular. And by 8 or 9 months, they become completely irregular. As you can see, each dot here is one organoid. It's the data from one organoid. As we increase in time, the variability of these organoids also increase, something that we don't fully understand, but it's seen. But on average, I can represent that with the population spike and voltage here. As I mentioned, in 2 months, we don't see anything. In 4 months, that's when we start to see the peaks of these oscillations. 6 months, they are there, very stereotyped. And by 8 months, they become highly complex. We start to see the overlap between those peaks, suggesting that they are moving from something that is early, rigid, to something that is more complex, abundant, and recurrent activity, as suggested here. So we are creating more opportunities for these neurons to communicate with other neurons in the cortex. And that's why the number of peaks that we see have this inverted U-shaped condition here, because the peaks start to overlap. So the oscillation peaks start to overlap and reduce the number. So this is a sign of a complex network evolving naturally, again, with no stimulation. They're doing that by their own. It's totally genetically encoded. We're not teaching them to do anything. And how similar are these oscillations with the human brain? So again, we are trying to compare something in vitro to a human fetal brain, where we have no way to probe electrodes in someone that is pregnant and said, can I touch the brains of your fetus? So that's totally unethical. So we have to rely on preterm neonatal brains. Sometimes when the babies are preterm, doctors recorded from their brains with these EEG caps to make sure that the brain is healthy. And something that is very characteristic of these preterm babies is the trace discontinuum on the EEG, which means that they have peaks of activity followed up by quiescent times. And as we grow older, the quiescent times become shorter and shorter and shorter, giving rise to a more complex EEG. So the LFP from the organoid has nothing to do with a more adult EEG, but is much more similar to the neonatal EEG. But how similar is that? And can that nature, can that response from the organoids evolve as human does? So we decided to do that using a brain machine learning algorithm. This is a regression model where we train the machine using about 500 EEGs from preterm babies that were born between 25 and 38 weeks. The machine can look at 2 or 3 EEG features and tell exactly the age of the brain, the age of the individual. So once the machine was fully trained, we feed the machine with the data coming from the organoid. So this is very unbiased. The machine doesn't know if it's analyzing the EEGs from the babies, the real human brain, or the signal from the organoid. And then we ask, can the machine guess right the age of the organoid? And the data was, it just blew me away when I saw that. What I'm showing here is the predicted age versus the actual age. And the perfect prediction would be here in the dot lines. The human brain data is in the black dots, and the organoid data is in blue. Every dot is a recording from one organoid. And you can see that, as I mentioned, is quite variable. It's not perfect. It's definitely more variable than the human brain, but it follows the same trajectory. And this is very significant. I mean, as they increase in age, it follows exactly the trajectory of the human brain, which is amazing that they're doing that by their own. Just a comment, the fact that the prediction is poor before 25 weeks is because we never had the opportunity to train the machine with data coming from the human brain on the first 25 weeks. We don't have the data. Nobody knows what happens electrically at the human brain in the first 25 weeks. So there is no way for us to train the machine. So we have a poor correlation here. But nonetheless, after 25 weeks, it's a very good correlation. And so that opens a new opportunity for us, as I mentioned, to start using these organoids to understand diseases. But we want to know if you can push that even further, if you can start to mimic or start to have oscillations that are more similar to a more adult human brain. And we thought that, well, at 9 months or 40 weeks, we don't have input and output coming from the exterior. So this is what happens when you're born. You start to open your eyes, you start to receive information from the environment. And we decided that maybe that's what's missing. Maybe these organoids are now anxious to receive some external input and process stimuli. So we decided to create ways to stimulate the organoids. And the visual system is quite interesting. I think Stuart mentioned several points about why the visual system is important for the consciousness studies. And here is our tentative to create a retina organoid. So this is from the same pluripotent stem cells. We create a retina organoid, a thalamic organoid, and the cortical organoid using our optimized protocol. These cells were engineered to carry reporters that label the photoreceptors in green and the ganglion projections in red, so we can see how the assembloid actually forms. It's kind of a Lego that we put them together, they fuse, and then slowly the axons from the retina passes through the thalamus and they start to find its target in the cortex. So we are following these structures now, but I want to show like one of the recent data that we have here in the bottom. It's the retina, and you can see the innervation of the ganglion neurons passing through the thalamus and reaching the cortex. So we are maturing these structures now, hoping that we can photo-stimulate the retina while recording from the cortex to see if we can create a more visual cortex, or if we can even create the identity of the cortex that would be similar to a more developed brain region. So this is one idea. The other idea was, okay, forget about mimicking human biology. What if we just stimulated these organoids using external stimuli that has nothing to do with the human biology? So this is our collaboration with CENI, the Center for Natural Intelligence here at UCSD. And it was started after I saw a movie from Star Trek where they have this brain from the monks living in this liquid that are connected to these machines, and the machines kind of are coordinated by the brains to move around. So we decided that, well, maybe we should use a brain-machine interface to create something to stimulate the organoids. So what you are seeing here is a robot that is learning how to walk using the electrical signal that's coming from a brain organoid inside our incubator. So it's captured by the multi-electrode array, and it's sent to a computer that feeds the robot with instructions wireless. So we are now stimulating these organoids by adding sensory information, such as these eyes. So these are infrared. As soon as the robot reaches an obstacle or a well, it stimulates back one of these electrodes. The networks rearrange. The computer now sees that there is a stimulus coming. There's a peak, there's a spike, and send a second command to the robot, such as stop and walk back. So in the future, we want to close this feedback loop and remove the computer from the middle and having the organoid controlling the robot autonomously, receiving the stimulus from the environment. So we want to know if the organoids can learn how to navigate a specific environment based on a previous training. So that's another thing, and we hope that these will stimulate even further these organoids to create new forms of oscillations. So the other thing that we thought about was, what if we age the organoids, just age them? And one way to age it is to grow organoids in microgravity. And we know that microgravity do affect the age of the cells. So we are growing some of these organoids at the International Space Station. This project was actually inspired by the NASA Twins project, where they show signs of cognitive decline in the astronauts that stays at the space station for long periods of time. At the cellular level, what happens is that the telomerase kind of get very short upon return to Earth, mimicking an accelerated aging. So that's exactly the experiments that we want to do, but instead of sending people over there, we're sending the organoids. So the organoids go in these specific boxes, and the only thing that the astronaut has to do is to plug the the platform for command inside one of these cabinets in the background. And then we have immediately access to these organoids. We can control how they grow. We can analyze them over time and then bring them back. So once they are back, the telomerase activity happens, and we don't know the mechanism. We don't know why they get stalled at the microgravity environment, and upon return, they just age. But the cells age together because of this telomerase activity. And our hope is that by doing that, we're going to age these neurons. They will mimic a more adult brain. We'll see if that's happening. This is ongoing. So, but all of that brings interesting questions. And I think I'm getting to the point of this seminar. So these organoids are not brains. This is a very nice article from Carl Zimmer from the New York Times, where he discussed some of these data. And of course, I mean, it raises some of the ethical questions about as we increase the complexity of these organoids, they get more and more closer to the human brain, or more and more closer to a conscious human brain, posing a conundrum for the science. I mean, should we continue to use this tool? Is there a threshold where we should stop? I mean, these are philosophical and ethical questions that we are trying to answer or trying to debate among ourselves. But there is one important thing, and Stuart was probably the first one who noticed that, well, I mean, this can be a really good model to understand the emergency of human consciousness in vitro. And so, and that's exactly the point here. So can we study brain organoids? Can we use them to study consciousness? So we were looking for consciousness theories that could perhaps be use this tool, and not all of them can be leveraged on brain organoids. But the point is, Orch OR could be, and this is Roger visited the lab just checking out some of these brain organoids. So that sparked a very nice collaboration with the Penrose Institute here in La Jolla, Stuart in Arizona, and my lab here in San Diego, where we decided to brainstorm potential experiments to actually generate or detect any level of consciousness using these organoids. So Stuart and I, we co-authored this very nice comprehensive article on how one use cerebral organoids for testing for consciousness. I invite you all to read. And in one of the graphics that he also shows here, the cerebral organoids might be in between a C. elegans and a mouse brain. So it contains all the layer 5 neurons, all the microtubule rich potential for these experiments. And we also propose kind of a guideline or stepwise schematic to test specific theories or to propose specific experiments. And I won't go into details. We discuss in lots of details in the article, but basically the idea is that some of these organoids do show some of the things that we believe are hallmarks of consciousness. For example, these high gamma EEG or the nested coupling EEG that we also see in the organoids. So there are other that we are about to test, but nonetheless, once we have those, you can potentially test for consciousness. And one way to do that is by perturb these organoids with specific chemicals, similar as we did with by cucuin or TTX. We can perturb the organoids while recording from them. And the idea would be to test with anesthesia, for example, to see if you can inhibit the emergency of these oscillations. And if we wash out the anesthesia, if the oscillations get back, similar to what happens into the human brain. So that would be strong evidence that there is some kind of consciousness level in there. And the same thing would be with psychedelics. I mean, we can increase or enhance these oscillations or this behavior to see if we can achieve a level that would be similar to what detected by EEG in humans. And of course, although we don't have strong evidence of a computing behavior in organoids yet, this is something that we are working with the Penrose Institute here. But nonetheless, we could use already some of these membrane protein inhibitors or microtubules inhibitors in the organoids to see how they behave. And the idea is to have a chart where we can see yes or no for most of these treatments to see if they support or not Orch OR. So it's not going to be like a definitive answer, but I think would put us close to a strong case for Orch OR. So at least it's testable, and that's why we like this hypothesis. Another thing that we are doing, one of the most difficult aspects of this research is to optimize the tools so we can do that in organoids. For example, anesthetics require a liquid-gas interface that we don't have currently working for the organoids. The other thing that we didn't have so far was a way to stimulate and record the organoids at the same time. And now we can stimulate these organoids chronically over time. So this is some of the organoids growing inside this plate that is being stimulated for several months now. So we're going to see if the stimulation can actually, for example, increase different frequencies in these oscillations. Lots of experimental details that needs to be worked out, but I definitely think these are all feasible, and we'll probably be able to do it in the next year or so. So I'll finish here by saying that brain organoids, they do show some similarities with the human brain. There are caveats, as I mentioned, but most importantly for us, it is these electrophysiological signatures that mimics these preterm neonatal humans, and it's possibility that we can enhance that. I would like to thank the funding agencies. My lab is mostly funded by NIMH and CERN, and just a full disclosure, I'm a co-founder of Tismo. This is a company that uses brain organoids to find better treatments for autism. And I mentioned lots of research from different members in my lab, but especially I'll give kudos to Kleber and Priscilla. This is a couple from Brazil as well who joined my lab, and they were tenacious enough to develop this new protocol that allow us to see the emergence of these oscillations. So without their persistence, we wouldn't be discussing this topic here. So thank you so much. I'll stop here.
All right, thank you, Allison. That was super cool. Such exciting stuff. You're putting organoids in space. Quite a trip. I'll take the first question. So there's a lot of genetic structure, you know, these brain nuclei and these white matter bundles. And, you know, being a cognitive neuroscientist myself, we often think about neural oscillations as arising from, you know, thalamocortical loops or specific, you know, brain regions that have, you know, central pattern generators or something. Yeah, to what degree the organoid, you know, lacking these definite structures, yeah, how do you really kind of cross that bridge into the structure that's inherent to the human brain?
Yeah, it's a great point. We think that perhaps these experiments should be performed not only with the cortical organoids, but with the thalamus. So we have this thalamic cortical loop, but also brainstem. We recently developed a protocol to create a brainstem where we can put in the trio as well. So if we can purposely create these structures and confirm that they are doing what they are supposed to do, I think we get closer to the circuitry that Stuart mentioned in the beginning. So that might be a way to do. Oh, especially if we have the retina capturing visual inputs, even better if we have that. But as you see, I mean, the complexity of working with a single organoid is already kind of high. By adding all of that, and each one has its unique developmental stage that you need to combine to be able to have something like that. So it is technically challenged, but it's not impossible. It can be done.
Yeah, super cool. Okay, there's a lot of people in the Q&A asking, do you have any ethical qualms with the potential for consciousness within the organoid?
Yeah, right now we have no evidence. Of course, as we do these experiments, we might be surprised. We might find that even in the cortex alone, we already have like some signs or evidence of consciousness. So we work together with the Center for Ethics in Biomedical Research here. We keep them updated of our data. ISSCR, the International Stem Cell Research Society, also oversees this kind of experiments. So we communicate if we have something different or unique. But I mean, I think it's gonna take a while to have like a definite proof. But let's suppose that the answer is yes. Let's suppose that, yeah, we have evidence that these organoids are now conscious or have some level of consciousness. What should we do? For research, this is not, at least in my perspective, not a big deal. The reason why is that we already use mouse for research who are animals with consciousness. But the human aspect might create like, oh, but these are humans. So if that's the case, we'll probably have to agree, we scientists, and together with the society, on a set of rules on how to deal with these organoids. How should we dispose them? How many should we grow for a specific experiment? What is a more humanistic way to grow these organoids? Do they feel pain if we add pain receptors to them? So these are all open questions, but yes, we are keeping an eye on the ethical question. It's a very important one.
All right, thank you, Dr. Muatri. I think that's all we have for questions. Super exciting. You're truly like at the front, like the forefront of neuroscience here. Very cool. All righty, so next up, this kind of concludes the long form portion of today's session. We'll be moving into the panel. And so I will not be introducing people from here on out, but I will introduce Dr. J.Tuszyn. One second, let me find my notes. All righty. Dr. Tuszyn currently holds the Allard Chair in Experimental Oncology at the Cross Cancer Institute, where he leads an interdisciplinary computational drug discovery group. Dr. Tuszyn has played a crucial role in the history of the Orch OR theory by working with Stuart, as Stuart mentioned, historically through many key early papers, to flesh out the specifics for how Orch OR can be realistically instantiated in microtubules. His expertise in the physical and chemical properties of microtubules is essential to the experimental validation of the Orch OR model. So let's welcome Dr. Tuszyn to lead the next panel.
Thank you very much, Justin. Hello, everybody. It's my great pleasure and honor to be hosting this panel discussion and presentations on the quantum biology of microtubules. I would like to join in the chorus of congratulations to Professor Penrose on his fantastic Nobel Prize in 2020, and also wish him all the very best on his 90th birthday. I also want to thank Stuart for organizing this wonderful event, very exciting and informative, with lots of interesting insights. And on this note, I would like to comment on Greg Scholes' talk, which was super interesting, and introducing the scale-free, self-organized type of model to the consciousness debate, I think is very valuable. But I would like to remind some of you that I think some similar ideas were expressed by Al Scott many years ago in his super interesting book, Stairway to the Mind, which I recommend to read. Obviously, it was not dedicated to quantum theories, but I think the merger of emergence and quantum is quite possible, as Greg indicated, and I tend to agree with this. Now, I want to say a few words about, since history was mentioned here, on the historical note, to introduce this panel. So I kind of look back to 1987 as the watershed moment when Stuart published his book, Ultimate Computing, that if you haven't read, you should, because it's an amazing document that I personally read through the night. I couldn't put it down. It was so fascinating and changed my life, my career as a scientist. I think it's as good today as it was in 1987. And then in 1991, I was honored to be a co-organizer of a workshop funded, by the way, by NATO that took place in Tucson, Arizona. And Stuart was a co-organizer, and the title was "What is Life?" And I think that was the beginning of this collective adventure. By the way, that coincided with the first Gulf War, and the sponsorship by NATO, I think, was probably coincidental, but maybe not. Now, we move fast forward to Roger Penrose's monumental books, 1990 and 1994, Emperor's New Mind and Shadows of the Mind. And I think that led to the awakening of sorts of the community to these tremendously interesting and complex issues. And then 1995, 96 is the proposal published by Roger Penrose and Stuart Hameroff on Orch OR. And then, so most of it until now, until recently, was quite theoretical. Maybe some of it was computational, a lot of work, a lot of papers. And of course, two of the Science of Consciousness conferences that Stuart organized from 1994 until now. So roughly a quarter century has passed until the moment that we are now looking at experimental validations or experimental falsification for some of these truly amazing ideas. And Alyson's talk was really groundbreaking in terms of where we can go with this. But I want to step back, and the title of this panel is Quantum Biology of Microtubules. Microtubules are made up of protein called tubulin. And in this connection, I think we'll hear some recent reports from several people involved in the project, which is funded by Templeton, started this year and going into the next on actually exactly this experimental testing or falsifying Orch OR from the ground up, that is looking at tubulin microtubules. Can they or cannot support quantum effects? And I want to add to this so maybe simplistic overview aspects, which I think are very important and probably chosen by nature, not by accident, namely the environment in which tubulin and microtubules exist, the requirements of pH and concentrations of ions, and the incredibly interesting architecture of microtubules. So all of this should make sense. And I think we're looking for demonstration of this. So we'll be hearing today from several people involved in this Templeton project about the electric, electrostatic, electrodynamic, electroconductive properties of tubulin microtubules, which are very unique in the world of proteins. And that protein tubulin and its assembly in the form of microtubules is probably not accidental, an achievement, evolutionary achievement that took place roughly 2 billion years ago, and has, so Mother Nature has maintained that and tweaked with this because pretty much all eukaryotic cells have microtubules, use them for various functions, and neurons, of course, have a special architecture of microtubules, quite distinct from dividing cells and other cells. So I'm not suspecting any randomness here, rather, I wouldn't say deliberate choice, but refinement through evolution and experimentation. So we have now the question about quantum, of course, is a big one, and hopefully we'll hear a few reports in this regard. We have 5 speakers lined up. The first one will be Aarat Kalra from Greg Scholes' group at Princeton, then Travis Craddock at Nova Southeastern University, he's associate professor there, and then Aristide Dogariu, professor of physics at the University of Central Florida, Bruce MacIver, professor of neurophysiology at Stanford, and A.Bandyopadhyay, senior scientist at the National Institute of Material Science in Japan, all of whom will give you the latest on our quest to find out the truth. So I think I have saved 2 minutes. I will then invite Aarat, who is the first speaker. Each speaker has about 10 minutes, but we also have half an hour dedicated to discussion. So we're not too nervous about time limits, but I recommend sticking to 10 minutes, maybe 12 if you need an extra slide or two. So with this introduction, I would like to ask Aarat to talk to us about his work on light at the end of the tunnel for microtubules, I guess.
Right, thank you, Jack, for that introduction. I am Aarat Kalra. I'm a scientist in the Scholes group. Professor Greg Scholes just gave a talk. I work to determine the feasibility and relevance of quantum effects within biological polymers, specifically microtubules, of course. So microtubules are these hollow cylindrical polymers. A microtubule is shown here. They are made up of a protein called tubulin. So tubulin is a globular protein, and it arranges in a linear head-tail fashion so as to form the cylinder that we call the microtubule. And of course, microtubules are present all across the cell. Now, classically speaking, microtubules play a variety of intracellular roles. They're critical for providing cell shape and rigidity. In fact, they, along with actin filaments and intermediate filaments, form what's called the cytoskeleton, which translates literally to the skeleton of the cell. They're necessary for cell division, and they basically act as tracks for the transport of macromolecules across the cell. So if you want to move mitochondria or any other membrane-bound organelle from point A to point B in the cell, it's done along a microtubule. You can see the motor protein almost walking on top of a microtubule. And so microtubules direct macromolecular transport to different places in the cell. Now, this is why biochemists and biologists are interested in microtubules. And a large amount of research is devoted to their biological function, such as the ones I've showed you here. This is not, however, why we're interested in microtubules. We're interested in microtubules because of two reasons, largely. The first one is that microtubules have an abundance of aromatic amino acids. Okay, so tubulin has phenylalanine, tyrosine, and tryptophan that allow it to interact with light. The second reason why we're interested in microtubules is that tubulin is arranged in a lattice formation inside a single microtubule. And that allows for long-range order within a biological cell. And so throughout this, this talk of mine, you'll see that the abundance of aromatic amino acids, one, and the lattice formation of tubulin within a microtubule, two, will continuously play up. So these are important points. If you look at the first point, which is the abundance of aromatic amino acids, so the three aromatic amino acids are tryptophan, tyrosine, and phenylalanine. Tubulin has 8 tryptophans, 40 tyrosines, and 40 phenylalanines. Now, bear in mind that this is just one isotype of tubulin that I've picked. So different types of tubulin will have slightly different numbers. But given that the volume of a tubulin dimer is about 240 nanometer cube, this abundance of aromatic amino acids is similar to that in chromophoric residues within light harvesting bacteria and photosynthetic complexes. So this abundance of chromophoric residues, and given that it's similar to that in photosynthetic complexes, has provoked the question of whether they could actually play functionally relevant roles within biology, as similar to photosynthetic complexes. Now, of course, photosynthetic complexes are exposed to light. Where are they found? On the surfaces of leaves. Even if it's an algal, even if you look at an algae, it'll be on a light exposed surface. So photosynthesis takes place in light-rich environments. And the obvious question is that if quantum effects and interactions with light are taking place in tubulin, it would have to be a light-rich environment, right? And in the brain, it's not entirely obvious where light would come into play, right? So, in a leaf, you have sunlight, it's a sunlight exposed surface, but inside the brain, where does the light come from? Well, the idea is that you see there are membrane bound organelles in the cell that release light. So if you look at a mitochondria, it emits these singlet oxygen species that then relax back. So they have singlet oxygens that gets excited, and when they relax back, they emit light in the visible regime. The idea is that tubulin and microtubules could absorb this light in the UV visible regime, allowing it to utilize the lattice-like structure inside a microtubule and transmit this optical information. And so the idea is that this is how long-range excitonic transport would take place via ultra-weak photon emission. So that is basically via light emitted through mitochondrial membranes. So in the Scholes group, this is one of the streams that we're interested in. The other stream that we're interested in is slightly unrelated, but not quite. So this is to do with intracellular computing. So of course, as I mentioned, tubulin is arranged in a lattice-like structure in a microtubule. The idea here is that upon the binding of a map to tubulin, so that's the purple blob here, the electron cloud in a tryptophan or a tyrosine or a phenylalanine would be shifted. And the propagation of the electron cloud along the microtubule lattice would allow it to be present in different patterns. So see, depending on where a map bound, how it and the isoform of tubulin and the exact GTP state, the nucleoside state, you'd form different patterns, like so. And so the idea is that intracellular computing could take place through automata in microtubules that would be generated through map binding and through excitonic propagation. All right, so this is the idea. Of course, Penrose and Hameroff take this one level further. They say that quantum computing could take place inside the cell. And so this is the second stream that we're interested in. The third stream that we're interested in is microtubules as targets for photobiomodulation. So if you have IR light and you expose it to a biological system, it's seen that IR light can actually cause benign effects. So this is a classic case the video that I'm showing you here, where optical pacing was achieved. So they could literally control how fast the heart beat in a quail embryo by the firing of IR light. Now, fascinating as this is, the intracellular mechanism by which this might take place is not well understood. So how does IR light exactly cause things like wound healing, neurostimulation, pain relief? While the effects are obvious, the mechanisms are not. And because microtubules are seen to reorient in the presence of IR light, and the famous work of Albrecht Weller showed that centrosomes might actually be involved in IR light photoreception, we think microtubules might actually be involved in IR light interactions with the cell. And of course, it's not just IR light. So if you have UV exposed tubulin, it's seen that microtubule formation using this tubulin is actually inhibited when it's exposed to UV light. So maybe I can show it to you over here. So on the left, we see a not UV exposed set of tubulin dimers. You can see that they form microtubules normally, but when you expose it to UV light, microtubule formation is inhibited. And in a plant cell, UVB radiation, so that's 280 to 300 nanometers exposure, causes microtubule network rearrangement. Again, the mechanisms are unclear. And so how light interacts with the cell via microtubules, or maybe without microtubules, this really forms the third stream that we're interested in. And so our aim is to, as I mentioned, gain a deeper understanding of how microtubules interact with light through these three streams: photobiomodulation, intercellular computing, and ultra-weak photon emission that would generate excitons within a cell. Open questions in the field. I've just listed some over here. how could specific substrate tubulin binding and unbinding be controlled? So this is how could you induce MAC binding to a specific site, causing a specific type of pattern to generate, and things like that. How is the output of intracellular programming converted to function? So what's the, how would automata cause a biochemical reaction to take place at the end of a microtubule, and so on and so forth. Some of these questions were given to me by Professor Greg Scholes, whose talk you just heard. With this, I'd like to thank you for your attention. We're very grateful for to the Templeton Foundation for funding this work. And I'd like to thank Professor Greg Scholes and the Templeton team and the Scholes group for helping me out in experiments in this direction. So thank you for listening.
Thank you very much, Aarat. Thank you, everybody. I think we'll wait with the questions until the end of the presentations by everybody in the panel. So we will now move to the next talk by Dr. Travis Craddock from Nova Southeastern University in Florida. And we'll hear about the presence of resonances in Raman spectra of tubulin and microtubules.
Okay, so my name is Travis Craddock. I'm an associate professor in the Department of Neuroscience, Computer Science, and Immunology at Nova Southeastern University in Fort Lauderdale, Florida. And I serve as a director of the Clinical Systems Biology Group in the Institute for Neuroimmune Medicine. And our main goals are trying to find novel diagnostics and treatments for any illness that involves a neuroinflammatory disorder. As we've heard from Stuart previously, that a lot of neurodegenerative disorders like Parkinson's disease and Alzheimer's disease, they have a dysfunctional microtubule cytoskeleton. And so part of my work is looking at the properties of microtubules and trying to find how to leverage those properties for treatments and diagnostics. I'd like to thank the organizers, as well as wish Sir Roger a very happy birthday and congratulate him on his recent Nobel Prize. I'd also like to thank him for providing a lot of the encouragement and motivation to get into this line of work. It's been truly monumental for me. I'd also like to thank the funding providers for the work that I'm about to provide or to discuss. Primarily, this has been funded by the Army Research Office, as well as from seed money through Nova Southeastern University's President Faculty Research and Development Grant. I'd also like to thank my collaborators on this project, Professor Alfano up at City College of New York, Professor Shi at UC San Diego, and Professor Galvez at Colgate University. So in this session, we've heard a lot about microtubules in general. Now, one thing about microtubules, because of their unique lattice properties, their hollow makeup, the hollow tube geometry, they're very unique nanostructures. And so trying to understand how these structures work, we always draw on similarities between materials that we understand in a more detailed manner. So one of these materials is a multi-walled carbon nanotube. So a multi-walled carbon nanotube is made up of sheets of graphene, pictured there on the far right. And you just wrap these carbon nanotubes around one another and you form these layers. And the size and the shape of these multi-walled carbon nanotubes can be quite similar to that of a microtubule pictured in panel A and B there. So multi-walled carbon nanotube can have an outer diameter ranging from 15 nanometers out to about 25 nanometers, which is roughly the diameter of the microtubule. Interestingly, these multi-walled carbon nanotubes possess similar properties to microtubules and assist in enhancing microtubule assembly and stabilization. So it's been one goal of bionanotechnology to mimic microtubules. Likewise, understanding carbon nanotubes can help us understand what the properties of microtubules are. And since carbon nanotubes can act as optoelectronic or quantum information devices, potentially microtubules can as well. Now, one of the ways that you can examine multi-walled carbon nanotubes is through a spectroscopy called Raman spectroscopy. And this involves sending light into a system, and it excites it from its ground state up to an electronic excited state, depending on the type of spectroscopy you're doing. And then when it relaxes, it doesn't relax down to its total ground state, but it goes to an excited vibrational state. So it excites vibrations within the system. And if you measure the difference, the shift in energy from the incoming light to the outcoming light, you can pick up which modes of vibration you're setting up within the system under investigation. So you can do this in other materials, but you can also do it in proteins as well. And so what you can find is where these modes of excitation are, but not only where they occur and what their intensity is, but the shape of the peaks that come out can also give you information on what's going on and how this system is behaving. So in normal processes, the shape of this peak is usually symmetric. It follows a Lorentzian or Gaussian shaped curve. However, if that vibrational mode, that discrete vibrational mode that gets set up within the system is coupled to the electronic modes of the system, the continuous electronic modes, then you don't get a symmetric system anymore. You get an asymmetric system. And this is referred to as a phonoresonance. And the degree of coupling determines the degree of asymmetry within the system. So you have this coupling or this Fano factor Q, which is related to the coupling strength. And the closer that value Q is to 0, the more asymmetric your term becomes. Now, if you make your Q value quite large, then you just return back to the Lorentzian shape. So this kind of phonoresonance has been observed in multi-volt carbon nanotubes. So we wanted to look and see if we could observe this type of behavior within a microtubule, which might give an indication that there's some degree of coupling between vibrational modes or phonon modes and electronic modes or exciton modes within the system. Now this process is, I just want to say, is 100% quantum mechanical. It's not a classical in any way. So finding evidence of this or confirming that you have some sort of phonoresonance occurring in microtubules is evidence that microtubules may be able to support quantum-like behavior or quantum behavior directly. These are the experimental methods. I'm not going to go into them in detail, but we did excite the system with a 532 nanometer diode laser and acquired these measures across a time of 70 nanoseconds. So initially we did this in dry powdered microtubules and then compared it to microtubules within solution. We used a MATLAB program to identify the peaks within the system. And doing so in both the wet and dry scenarios, we found 24 main peaks within the system with slight variation in peak location between the wet and dry solution. Now, the main peaks of these found within microtubules can be attributed to the aromatic amino acids, phenylalanine, tyrosine, and tryptophan, as well as disulfide bonds within the system. There are a few other peaks around the amide 3 vibrations, as well as some valine and arginine peaks. But for the majority, it's around these aromatic amino acids and disulfide bonds. So these are the actual spectra that were obtained from the microtubule samples. And in order to try and define, are these symmetric or asymmetric, what is typically done is you highlight the peak of interest and look at it within a window. However, what this does is it neglects the effect of neighboring peaks. And so if you have two Gaussian or Lorentzian waveforms side by side, but you don't include that neighboring peak within your analysis, what may appear as an asymmetry may just be the fact that you have two Gaussians close together. So what we wanted to do is we wanted to fit the entire spectra all at once. So what we did was we applied a Fano function to each individual peak that we found, and then optimized all of the parameters within the system and evaluated the parameters of the system and its agreement with experiment using the Ikeki information criterion, which is a method to determine whether your current model is better or worse than your new model. And so by minimizing this value, you can find out which is the best model to describe your fit overall. And so we did that over several thousand, 10,000 iterations, 40,000 iterations to try and find the best possible fit between our experimental data with this theoretical modeling fit. And so what we found is that within the wet microtubules, within the dry microtubules, we only found one potential location of phaneroresonance. But within the wet aqueous solution, we found 10 possible phaneroresonance behaviors, which includes that one that is found in the dry case. So I'm just going to discuss the wet case for now. So what I'm showing there is a blow up of the individual peaks that comprise together make up the experimental model fit. So each of those peaks there together, when you sum them up together, give that fit that you see above. So experiment is shown in red, which you can barely see because the model fit shown in black traces it so well. So we defined anything that had a parameter, that phanoparameter Q of less than 10, well, between 10 and minus 10, to be indicative of potential phaneroresonance behavior. And what we found was primarily that these phaneroresonance behaviors, or potential phaneroresonance behaviors, are within the disulfide bonds, as well as within tryptophan or tyrosine within the system. And that's for both the low wave number region, as well as in the high wave number region. Now, this isn't conclusive proof that this is phaneroresonant. There may be other methods or other effects that are taking place to describe this, but they are phano-like line shapes. So this is something we're further investigating, but it is a potential avenue or experimental proof that there may be quantum effects going on within microtubules, evidence between the coupling between phonon modes and exciton modes within the system. And since I'm out of time, I'll leave it there.
Thank you very much. I'm itching to ask questions, but I think I'll have to wait until the end of this panel. Like everybody else, you're slightly over time, but not much. So we will now move on to the third speaker, Dr. Dogariu from the University of Central Florida, and he will be talking about remarkable electromagnetic properties of microtubules.
So first of all, thank you very much for the invitation to be part of this unique celebration. It is both a pleasure and an honor to be sharing some of these results that we collected over the years regarding topics that are tangentially pertinent to a lot of the things that we have heard here before. So I must confess from the start that we really did not conspire to make these topics complementary, but it just happens that this one will certainly complement the previous talks. So what I'm going to summarize here are some aspects in which collective phenomena are manifesting and are measurable at mesoscopic scales. One of these instances being this microtubules that we have heard a lot so far, and I will try to connect these observations to the specific structures that these systems have. So first of all, let me just make very clear what I mean by cooperative effects. It's everything that is opposed to independent interaction, right? So if we have a system, irrespective of its scale, it can be decomposed to elementary units, which can contribute independently to a given result, or they can act collectively and produce a result that depends directly on the level of interaction and the internal structure of the system. You will see here some examples that relate to seemingly random structures, but I just want to make the point that This seemingly random appearance of many of these structures is probably deceiving at some levels, because nothing is really random at all scales. And this is true in both time and space. The first example I want to show you is the local density of states in a system in which the same amount of matter is distributed differently, right? The density of states, which is probably not unexpected to really manifest different type of distributions when this level of cooperativity, which is driven by the local structure, changes. Another example, also a macroscopic example, a global manifestation of this cooperativity. I have another example here in which I take exactly the same amount of matter and redistribute it. And then when the interaction between the elementary units gets stronger and stronger, new eigenchannels of propagation open. If you want to use this language, an additional propagation channel of energy that really relies on evanescent couplings opens up, and as a result, surprisingly, the more you compact the same system, the more transparent it becomes, practically. So these new eigenchannels in electromagnetic interaction here appear just because of the very strong and unique structure-dependent collective interaction that happens between elementary units. Another example, there are in propagation of electromagnetic fields through systems, these competing mechanisms, which could be, perceived as exchanges of real or virtual photons, if you wish, they appear to be dependent on the level of macroscopic interactions. In other words, the deeper you go into the medium, the more you're gonna uncover these kind of anomalous phenomena in which the radiation becomes localized. It just cannot propagate. And practically the transport of energy is done on a time-dependent random potential, which is really similar to how we describe the wavefront and the time-dependent Schrödinger equations. Another manifestation is the non-stationary statistics of these kind of waves. All of them really relying on the specific structure. Now, if you go into more organized type of systems, in quasi-structured systems, and just want to make the point here to remind you of another contribution of Sir Roger Penrose about the quasi crystals that actually he opened up a whole new area of research. This is a manifestation that is very close to that regime of interaction. When the system is driven close to a pseudo band gap, that means that there is just the appropriate mixture of order and macroscopic disorder. When you are approaching the conditions for which this Anderson localization is supposed to occur, then the interaction is practically carrying this fingerprint of scaling with the volume of interaction with the medium. Another example that we've been working for quite some time now, actually in evaluating quantitatively the level of cooperativity in scaleless media, right? In which you have a system. This is a real fractal system, which, like any kind of real system, is practically described by two scales, right? A scaling parameter of the structure and certainly an extent of this scaling law. Then you find that different signatures, different type of signatures. This is actually the shape of this enhanced backscattering cone, which is a specific manifestation of weak localization. That carries, again, the fingerprint of the specific structure of the medium. So far, I think we showed examples that the structure really matters at macroscopic scales. Now let's come back to the structure that which is the subject of our discussion here. So this structure, it's a helical system in which one could conduct a quasi-static analysis of all the possible vibrational modes, depending upon how you model this structure. I'm not going to get into any details of the model, but I will just mention some of the outcomes of it. So, of course, because of this highly regular structure, there will be resonant frequencies. But most importantly, the coupling between an emitting dipole in the proximity of this structure will have a non-local coupling to the structure, will be delocalized, and will mediate long-range dipole-dipole interaction in the proximity of this. So that would be equivalent to an enhanced range for a FRET type of energy transfer in the neighborhood of this microtubule. Certainly, it will modify the local density of states here, leading to enhanced Purcell factors, leading to an entire host of potential quantum effects, just because of these localized and localized resonances and delocalized excitation that happens in these structures. But everything that we talked so far is essentially what I would call in steady state temporal regime. Now let's take a quick look onto something even more interesting. Let's look at non-stationary phenomena, and the subject has been touched before, and look at how the dynamics of the emission from a system that is structured, how that dynamics depends on the temporal profile of the excitation. So let's suppose that I have a medium that is excited by a very short pulse. And then at the end of this short pulse, practically all of these elements of the system becomes secondary sources. And all of them are actually treated in the same way, right? So they will be essentially emitting in the same way. But now let's consider another situation in which first, the system is charged, it's excited at steady state, and then I remove the excitation abruptly. The structure in both sides is exactly the same. However, when I do the second one, because I pre-charged the system, the emitters, the secondary sources, will start emitting with different amplitudes and different phases, even though the structural composition is exactly the same. So we have practically different initial conditions for emission, even though the structure is exactly the same. So how we excite this really matters. So what? These are now oscillators which are out of phase, and they oscillate with different amplitudes. Certainly, they will eventually, because of this cooperativity between them, they will, for lack of a better word, synchronize. right? Even though they were initially out of phase. This collective states would lead to, at short times, to what is called superradiance, or at very long times, subradiant quasi-modes of emission. The difference between these two regimes of emission and this dashed black line is essentially the decay rate of an independent oscillator driven for a certain shift. So in time, certainly the cooperativity manifests in two ways. A lot of attention is devoted to the superradiance because probably it's just the prefix super that everything is good when it's super. But the most important, some important things are also happening here, because here it means that light comes out of the system for a very, very, very long time. But there is something more to it. At very short time, the superradiance points forward with respect to the excitation, while later on, the memory of the initial excitation is still maintained, even after a very, very long time. So even though the emission is subradiant, somehow the energy it's emitted in a direction that is remembering somehow how the light has been injected into the system. I'm not going to get into details of this, but we have an experimental study of this finding. Coming back to the system under investigation here. So these collective effects in this structure manifest from the level of a monomer to the level of the entire microtubule, to the length of the entire microtubule. The field is structured according to the size, the number of wraps, the angles, and so. And similarly, even at larger scales. So these kind of structural effects happen from the monomers to much larger scales. Even in a paired centriole, if you look at how the field is distributed in here with respect to the initial polarization, you will discover very quickly that you are looking actually at what we call an L-shaped antenna, which has the very appealing feature that is a broadband and is tunable. And guess what tunes the frequency of this L-shaped antenna? Is exactly this gap that with my formation, I'm just starting to learn now what's the meaning of separation between these centrioles. But certainly, this is a plausible mechanism for what has been suggested to be behind this rudimentary form of cellular vision by Albert Bührer. So,
Aristide, sorry, I have to cut you off. If you can finish in a minute or so, because
I have 1 more minute, 2 more minutes. So the subradiance in the microtubule has interesting features both in the time domain and in the spatial domain. I just want to show you here that the space-time fingerprints depend on the size of the microtubule. This is the emission patterns for different sizes. And these emission patterns really changes in time. So this system, if it's charged, can provide a space-time encoding of its location if it's in a system that relates to that. Now, the last slide that I wanted to show you is how these things actually behave in time. These are experiments of what is called in some circles delayed luminescence. We can also call it subradiance, measurements of subradiance in these systems. And just to show you that essentially we identify longer lifetime and stronger emission efficiencies for systems that are structured in this manner, in these spiral almost crystalline structures that produce these non-local space-time fingerprints. And to emphasize what Jack was saying, there are other type of the experiments that really point to a unique electromagnetic signatures that is imposed by these structures. And we'll be happy to answer any questions you might have. All right, I'm done. Sorry.
Thank you, Aristide. No,
but I heard that we had some time provision. So
yes, we do. Thank you so much. It's fascinating. I wish we could have half an hour just for your talk or more. You've really touched on some incredibly interesting stuff. So the next speaker is Dr. Bruce MacIver, Professor of Neurophysiology at Stanford in the Department of Anesthesiology. Bruce, are you there?
So keeping with the Templeton microtubule theme here, Rajneesh Khanna's lab and my group, both at Stanford, wanted to develop a system that we could directly visualize microtubule actions in real time in a living system. To do that, we've moved from animal brains to plants because they afford a very high resolution view of microtubule dynamic reorganization, and in particular, the phenomena of phototropism, that's where plants bend towards light, allows us a system to directly test whether microtubule organization could be sensitive to volatile anesthetics in the concentration range that we know produces a loss of consciousness in people. So to that end, we're looking at the growth of seedlings, and that's shown in these series of photographs. On the left-hand side, this column here shows in the presence of the volatile general anesthetic isoflurane at varying concentrations from none to surgical levels of anesthetic, we can in fact dramatically impact phototropism. Now, phototropism is the plants growing up normally towards the light that's above the plant until we turn on a strong blue light that we direct perpendicular to that growth. And as you can see, the plants bend towards that light. If we remove the isoflurane, we can continue to follow this, and you can see that they start growing back in the correct direction. But even at these low concentrations of half volume %, we start to impair the phototropism. You can see this is shorter here than in the control condition. So why is this interesting? Well, we know that this bending, the phototropism, involves the disassembly and reassembly of microtubules, and I'll show you that in a minute. At the highest concentrations, you can see that we completely inhibit growth for quite a while, but by the next day, all of these plants recover and start growing normally, so it's quite reversible. We're not the first to show that anesthetics can affect plants. Back in the late 1800s, Claude Bernard showed similar effects with diethyl ether and leaf movement in response to, excuse me, to touch. So how does this work? Well, we know the blue light causes the normal arrangement of microtubules. These horizontal blue lines represent microtubules, and this is an actual image of microtubules in one of those growing stems. When you hit it with blue light, it rapidly disorganizes and starts to bend towards the light. And by the time it's fully bent towards the light, the microtubules completely rearrange in now a parallel manner versus the perpendicular manner. So we think this could be an ideal system to look at the effects of anesthetics on microtubule organization, and that we can look at that in great detail. So to start with, we just have started to ask, well, where are the critical concentrations? And as you can see, it's somewhere between 0.5 volume percent and 3/4 volume percent, where you dramatically start to change the phototropism to the point where you can virtually abolish it. And then at higher concentrations, like we use in people to put them asleep for surgery, we stunt the growth. But again, this is all quite reversible, except for these unfortunate individuals here who are no longer with us. So what we're hoping with this system is that we can look at how microtubule dynamics are affected by anesthetics. We can study energy levels, growth signaling in very high resolution. And this has been done without anesthetics in these plants for a number of years now. It's quite a neat little model because we've got mutants of these plants that have known growth alterations. We can play with them. We can look at the blue light signaling molecules and the pathways. So we can get a very detailed look at what anesthetics are doing to cause this block of phototropism. Interestingly, some of the same target proteins that we know are in our brains that are affected by anesthetics, such as GABA receptors and potassium channels, are also used by these plants to, in their signaling pathways and growth pathways to facilitate this phototropic effect. Now, in the interest of time, I'll just end there and we can move on with our session.
Great, thank you very much, Bruce. I'm sure there'll be some questions in a few minutes. So now, last but not least, A.Bandyopadhyay will be speaking to us from Japan. He's at the National Institute of Material Science in Tsukuba. And he will be talking about quantum optics and microwave studies of tubule and microtubules, looking for fractal resonances in neurons. So Anirban, the floor is yours. We are keenly expecting your talk. I'm sure it'll be interesting. Thank you.
Thank you, Jack. Can you see the?
Yes, yes, we can, and we can hear you. We can also see you. And
okay. So this is a 2016 work. First of all, I would like to say happy birthday to Roger, and then Stuart. Thank you, Stuart, for organizing this, and thank you, Jack. And I would like to thank all of you who are present here. So this is a 2016 work, and then scanning tunneling microscopy. So it was discovered, this machine was invented in Japan in 1995. And then this machine allows you to look deep inside a biological system and without touching it. So you send the electromagnetic wave and then you excite the system and you can image what is happening inside. So, let's look into deep into that. So it's a, you can see in your, you have three panels, okay? And then left-hand side is a cultured neural network in our laboratory. And then in the middle, in the kilohertz domain, frequency domain, we are imaging how ions are moving and then our density. And to the right, we have dielectric resonance of the internal structures of neuron. So the filaments in the megahertz and nearly gigahertz regime, we can see that there are several events that is taking place. Different components are resonating before the neuron fires. So in 2016, we argued that ion channel image that you know for 100 years is not the absolute picture. There are filaments inside which directly affects the neuron firing. So that means the foundation of AI, artificial intelligence, and other things that you are saying is not true. So since 2016, we have published a series of papers only supporting the claim related to this particular event. So today, because of short time, you can go through our publications, and then you can check the results. That ionic picture is not everything. Deep inside the neuron, the silent neurons are firing. Silent filaments are firing. So then let's. This is your brain cell, which are firing. You can see tick, 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 tick sounds. And then this is microtubule, which is vibrating in kilohertz, megahertz, and gigahertz domain, different frequencies that are tubulin. So we carried out in the last decade, more than a decade, we carried out and published several papers on the resonance frequencies and then this is the sonification of the triplet resonance band of axon initial segment, which remove the membrane, and then we measured the filamentary bundles. So this is also from 2017 paper where we published that inside a single neuron firing peak, actually you can listen to the triplet resonance band, which are vibrating in an integrated fashion. So this is our latest paper in symmetry, and that is, I would like to highlight it today. So in the neuron, the same 2016 result, we started publishing continuously in different modes. In the top panel figure, you see kilohertz, megahertz, and then gigahertz domain. So what you can see, there is a ring separated electromagnetic field deep inside the action, just below the membrane. So there is a ring, electromagnetic field ring, like a vortex forms when the neuron fires. So we wanted to understand it. So I changed my laboratory from 2016. We started shining microwave or radio wave to the neuron, and then exciting particular component that is inside the neuron, like microtubule and different protein complexes, actin or others. We can purchase them from the market and measure the resonance. And we published it 2013, 2014. What we did from 2016 onwards is setting up a laboratory where we will not make any physical contact with the neuron. We will wirelessly trigger the resonance of the microtubular structures and send monochromatic photon and let it rotate the entangled photon. We will see how it comes out. So setup is something like this. Patch clamp is there, like normal studies. We shine with vortex lens the rotating photons or entangled photons, monochromatic entangled photons on the structure. And at the same time, with an antenna, with a precise frequency, we excite the particular component and then try to see who is doing what. So it's a beautiful result. So this is the schematic. So laser comes, it's on the sample with an antenna, we excite it. So this is all from the symmetry paper published 2 or 3 months back in the month of April or May. So what happens is entangled photons, actually photon rotates and creates a ring of light or photon. So what happens is with the helical structures or helical geometries, the geometric phase is captured by these light rings, and then they come up and then project like a laser hologram. So for membrane separately, we take out the membrane, we find light rings similarly like this for different structures, because reviewer was particularly interested that you have to deconvolute and you have to understand each and every light ring separately. So it was a very difficult time with the reviewers before the paper got published, because he wanted to understand whole thing first, and then we will allow your paper to be published. So microtubule neurofilament part, we found out its own optical quantum optic signature. So basically, light is coming, and then the ring you are seeing, it is actually one photon. It is not many photons. It is one photon, but it is rotating. And this is the probabilistic distribution. And you know that in 2018, for this particular discovery, that rotating photons, Nobel Prize was given. But this work, we use the technique of rotating photon to understand the different kind of structural parameters. So beta spectrin, actin, tubulin. So for different materials, we did it. We found the grammar of truth table first, and then the whole neuron. These are the pictures that you are seeing where with microwave, we excite particular part. You see the arrow? You will see some of the light rings are bright, some of the light rings are dark. Basically, this is from the symmetry paper. This is the figure 4 of the paper. So you can get it online, it's free. And so you can identify particular component, how it is resonating and how it is doing what. So basically we found that our latest technology is that with microwave or radio, we resonate wirelessly with an antenna, a particular component inside the neuron. And then with quantum optics, we can measure what is happening there. And then we could correlate that everything from the molecular scale to the largest neuron scale, everything is integrated with geometric phase. So geometric phase structure or architecture of geometric phase is important. In quantum mechanics, we study only one geometric phase, but we argue, we do not understand it right now, but we argue that some people, if they want to understand the consciousness or maybe understanding the brain mechanism, they have to find out the architecture of geometric phase. So basically, for 100 years, it was believed that neuron membrane is alone conducting, and then the ion channels come to the soma, and then soma bursts, and then an electrical vortex or the circular vortex comes. So this is from our 2016. We challenged this for the first time in the world, and we are continuing to do this. With this filament, we have done theory and experiment that this all these microtubule and neurofilaments and others, they integrate and they produce paired of vortices which interact, and all these results are obtained by quantum optics and microwave physics directly. And from the symmetry paper, you can find all these pictures are there. So these pair of electrical vortices are created which controls it. So basically, there is a hidden phenomenon that is controlling the precise timing. So humans' brain cognition actually depends. Brain cognition depends on precise fine tuning of the neuron firing. So millisecond firing is there, but if it is not fine tuned, the cognition will disappear. So that's what we are providing, that there is an alternate mechanism that is high resolution, temporal resolution control, and that is provided by filaments. And we are continuing that. So this is from the old result, transmission electron microscope studies and transmission electron microscope studies and quantum tunneling studies, where different kind of grid, protein grids on the surface of microtubule, lattice-like structures are generated at resonance. So what you do is you pump with electromagnetic wave wirelessly with an antenna, and simultaneously you measure the quantum tunneling or transmission electron microscopy. So what I have done in the last 10 years is that combining completely different kind of tools together in our laboratory. So what we do, we wirelessly pump, and then similar thing with this one. These were published in 2015. So you can see our 2015 papers by Sahu, and then you can find this quantum tunneling images, and then how they dance at different resonance frequencies when we pump them. So you can find out the frequencies and then how do they do it. So we did it with monomers also, dimer and monomer. So these are Stuart showed this result. So initially we were interested only in the triplet resonance band because we found that the resonance peaks actually change themselves, group themselves, and it's lifelike. So initially we did not understand. This was published in Fractal Fractional. So there is a fractal behavior from tubulin, microtubule neuron. I think it should be scaled all the way up, and there should be a brain model. So last 10 years, we developed a brain model, and this result you can see. We are going to build one of the largest databases, brain databases of clock-like architecture or clock-like behavior of entire brain body component, where you can see that all are connected with many, not just triplet of doublet of triplet electromagnetic resonance band together. So in future, what are we going to do? This is our brain model. In future, what are we going to do? is that we just purchased an instrument to look at the geometric phase architecture. So what we found, microtubule is unique because it does not follow the block sphere-like structure. It has a phase structure in the central region, in the Hamiltonian. If you look at it, it has a term called 3 cos theta, sum of cos theta, 4 pi of cos theta. So if you plot it, you will find it's a phase sphere with 12 holes, singularities. So we want to send 12 entangled photons into the microtubule and simultaneously see 12 integrated coincidence counter how entangled photons are behaving. So this experiment is not done, but we just purchased the instrument. In future, we are going to see. So my take for consciousness or understanding consciousness or biological system is to first understand how the geometric phases, many geometric phases are integrated together in an invariant structure. If we understand that, then we will be able to understand how the biological system is integrating information. So in quantum mechanics, we know the mystery of only one geometric phase. So I personally think that some theoreticians or some intelligent minds should come together and try to put many geometric phases, at least 12 geometric phases together, and try to create a scale invariant structure. Because you see, inside a tubulin protein, there is the alpha helix. It's a helical structure. If you go to the microtubule, it's a helical structure. Even a neuron, around it, there are glial cells in a helical pattern. Why helical shapes are always coming up? Because it can create a scale-free geometric phase structures. So today's discussion was that we will be able to understand quantum. We have to go beyond quantum, or we have to understand even the quantum phenomena properly. My suggestion is, I have not understood it properly. I'm trying to understand it, but geometric phase structure should be the way to go forward. Thank you very much.
Great. Well, thank you very much, Anirban, and thank you to all the panelists. Now we have about 20 minutes, just under 20 minutes left for discussion. I received a lot of comments from the audience. As moderator, I will take this prerogative of mine to maybe single out some of the panelists for questions, or maybe you can jump in if you wish. So one very intriguing question came from somebody in the audience, anonymous attendee, who said, what mental informational functions are enabled if there is a QM, quantum mechanics based order in the brain? I'll hazard an answer, which is actually quite new to me, and maybe there are some additional aspects, but Mike Levin from Tufts University in Boston and Chris Fields recently wrote a paper arguing that without quantum mechanics, the brain doesn't have sufficient metabolic energy to process all the information and perform mental functions. So this is my quick answer to this question, that actually quantum mechanics allows cheap information processing compared to classical methods of information processing in the brain. Would anybody else wish to comment on this question?
Consciousness.
Yeah, consciousness. Okay, all right. In one word, consciousness. So Stuart, since you are there, I just wanted you to address the issue that Sir Roger actually posed and people are still asking about, namely, what's the main difference between quantum chemistry, which is basically everywhere in all the molecules, and quantum mechanics in the context of consciousness? What's the main difference between these two concepts? Do you have a quick answer to that?
Well, we're talking about some kind of collective quantum effects, cooperative, and again, E sub G equals H bar over T, or the other way around, captures it, because this whole thing. Yesterday there were talks about quantum mechanics and general relativity don't jive, but I think Roger's connection to the two by consciousness, by this E equals H over T, kind of bridges between the two and takes care of the scale problems and the energy problems, because it's an edge. It's an edge, that actually slices through the whole universe. In fact, you can be very high E sub G and low T, and vice versa, depending on where you are. So it's a collective effect, and objective reduction is a collective effect. All the components have to be entangled and in superposition together for a period of time to reach thresholds. So probably other quantum phenomena, condensation and I don't know about entanglement, but are probably also collective effects that you only see at higher orders.
Sure. And yes, I would add to it long range, right? So this is collective long range. Quantum chemistry typically is atom to atom forming a molecule.
Yes, definitely.
So I have a specific question to Travis from the audience, actually, because our paper was mentioned and you were the principal investigator on this. Namely, can you link what was said today to quantum memory storage in, let's say, through microtubules? Is it quantum? Maybe not, but this issue of memory has not been addressed, but I think somebody would like to know if there's a connection.
I mean, we've come up with a couple of mechanisms. The one that I think you're talking about is the KMK2 phosphorylation patterns on microtubules. So the way that I would foresee that kind of linking up with, I mean, that's kind of a classical way of encoding memory. So you're making post-translational modifications to the surface of the microtubule, straightforward mechanical change. But as you heard from Aarat when they were talking about the way in which the, for example, exciton propagation may change on the surface of the microtubule due to maps being attached to the surface, those purely mechanical changes can then be changed into changes in excitonic patterns. And so we would expect the same thing to happen for these post-translational modifications, that they could modify that pattern overall.
Okay, great. Thank you, Travis. I have a question for Bruce. Actually, one of this is from the audience, one is from me. That was a fascinating talk, by the way. I would like to hear more about phototropism. The first question is, the anesthetic concentrations used in these experiments you described, do they correspond to those used in human anesthesia applications? And my comment, and perhaps you can relate to it, is something called gravitropism and microtubules. Very few people know about these experiments performed in the 1990s in space. The microtubules actually were affected by the absence of gravity, and they were not polymerizing and stabilizing the same way as on the surface of the Earth. So there could be a connection between gravity and photo effects. So, over to you, Bruce.
Yeah, well, I can't comment on the gravity aspects because I'm firmly rooted on the planet Earth, a big gravitational field. But in terms of the concentrations that we used, we purposefully chose the range that's used in people clinically. And the concentrations that block phototropism are very similar to the concentrate. So this is 0.75 volume percent, is right on top of the concentrations where people lose their ability to respond to verbal command. So they can no longer tell you their name. You're asking them to squeeze your finger, they stop squeezing your finger. So these are behavioral measures of consciousness that are routinely used clinically. Of course, we have no way of probing whether we're actually blocking consciousness itself, because some people have demonstrated that they've been aware even at surgical concentrations that are 1 volume percent and higher. But that's very rare. 99% of people have no recollection following surgery and can't recall any episodes that occurred when they're exposed even to 0.75 volume percent. So we were targeting concentrations that we know are associated with consciousness in humans and animals. And we're surprised to see that the phototropic effect is blocked by these very same concentrations.
Great, thank you very
Chris,
much.
can I ask? Oh, I'm sorry.
Go ahead, go ahead.
Do we know the effect is mediated by microtubules? And if so, is the effect on the assembly disassembly, or is the effect on the detection of the photons itself? Do the microtubules detect the photons? What's detecting them and orienting the system?
Well, this system, as I mentioned, has been very well characterized, Stuart. We know that there are a number of proteins involved in the phototropism, starting with proteins that actually sense the photons and convert the photonic stimulation into biochemical cascades, the typical cellular pathways, signaling pathways that end up at the microtubules. So to answer your question, no, we don't know that the anesthetic is directly affecting the microtubule to block this effect, but we have all the tools we need to find out. So we can trace it back step by step and see, it could be just as simple as the anesthetic shut down mitochondrial energy production, and without ATP, you don't do anything as a cell. So, but at the moment, we don't know, but we can find out. And over the next few months, we will find out.
Great, thank you, Bruce. If I may ask now, Anirban, maybe Aristide too. There was a question about the holographic theory, and I think Stuart mentioned this. And Anirban, you specifically mentioned the membranes and things of this nature were interacting with microtubules. Would you care to comment about the emergence of holographic images using microtubules? Have you thought about this? Is this part of your...
So what we did is a simple experiment. If there is a helical structure or there is a geometric phase generator, any device, if the photon falls on it, then that structure rotates the photon. So it generates a hologram. It projects the hologram. So what we did is a microtubule, you can take it out and you can measure the electromagnetic resonance. So this is going on since 1930s. People have been measuring protein resonance for a long period of time. So there is nothing new in it. What we did is combination of quantum optics and microwave. So microwave we shine or radio wave we shine on the neuron, on microtubule or other structures. And then when photon falls on it, photon comes out. But photon, when it comes out, photon interacts with different structural symmetries differently. For an example, if there is a water structure inside the microtubule, they interact with it differently, and protein structure differently. And if there are any other structures present in the microtubule, they will act differently. So we get many different distinct rings of light because of that. So this is how the hologram is produced from different things. So when neuron is shined, many components are there for the neurons. So how do I know what are the holograms belong to whom? So what we do is with an antenna, we excite microtubule or actin or other, and those holograms actually becomes brightened. They get brighter. So we understand, okay, they belong to them. So we can deconvolute this hologram. So we think that somehow my concern always have been that a neuron, we always say nerve spike is a one spike. Actually, that is not true. Nerve spike is a circular electric ring. It is electrical vortex. If you imagine membrane at action, it is a circular electric field. So that which goes on. So it's a vortex. It's a vortex. So I think that somehow vortex and holographic projection, electrical, ionic or polarionic or different things would be related. My work is very limited, but I think that others can comment on the vortex and other related parts and how that could be related to hologram.
Okay, thanks, Anirban. I wanted to turn to Aristide for a moment. Maybe you can comment on the hologram, but specifically, there was a question about quantum and non-quantum electromagnetic effects. I think your work touches on both. Can you discuss, so delineate, what's quantum, what's not quantum?
Yeah, just a point of clarification. the terminology again. So this, if we are talking about what we call holographic, in my understanding, that means a very extended phase correlation for the electromagnetic field. So in this context, there were some statement before. I think Greg made it, and I cannot but strongly agree with that. That synchronization, I think that these were his words, synchronization trumps decoherence. So that's absolutely true. So then talking about this kind of a phase synchronization that could be labeled as holographic something, you know, must be done in some kind of a temporal framework. So this is what we would need to actually clarify in the future.
I see.
Now,
so some sort of clocking is needed and synchronization across the brain, I guess?
Yeah, yes. We are not talking about a coherent phenomenon, you know, coming out of a laser, in which somebody else does the synchronization within the resonator. So there must be something there that actually could provide this reference for a holographic, you know, behavior. Right, now about the other part with what is normal electrodynamics and what is quantum electrodynamics. You know, at these dimensions, everything that we discuss is essentially quantum electrodynamics, because we are discussing, and all of these calculations that we have and the experiments we are performing are on structures which are significantly smaller than a wavelength, at least in some dimensionalities, right? So then absolutely all of these effects are of that nature. That doesn't mean that the manifestations cannot be detectable at mesoscopic scales, but the subtle interpretation certainly needs to go back to a quantum electrodynamic description. All of these virtual exchange of photons and things like that I was talking about, this is QED.
Tablet containing a portion.
Yeah.
Sorry.
Back to you.
Yeah, great. Well, since we're talking about electromagnetic effects, I wanted to ask, actually, Aarat, the last person that, the first to speak, the last to answer a question. So we talk about electromagnetic effects, quanta, photons. Maybe Aarat, and you talked about this in your talk directly, effects of photons on transformations in microtubules and ensembles. Do you know, and can you comment on the intensity of intracellular photon generation vis-a-vis these effects? Because obviously you introduced them from the outside and you talked about some experiments. Can you tell us about the relative intensities?
Yes. So the intensities, according to a paper by Devaraj in 1998, are of the order of 10 to the power minus 13 watts per centimeter square. So which is pretty low, right? So that's why they're called ultra weak photon emissions. Yes, I think very little work has been done on understanding how organelles inside cells would respond to such ultra weak emission. So that's I think one of the topics that we're interested in analyzing, and this speaks to what Professor Dogariu and what Professor MacIver found, you know, with the microtubules reorienting in response to light and things like that. So I think Professor Meissner had a question for Professor Scholes.
Yes, indeed. Yeah. There was a question about nonlinearity, and I think that Professor Scholes may be there.
Yeah, I'm here.
Yeah. So may I ask the question? Yeah, okay. So my question is related to the remark of Roger that quantum mechanics is linear and you have described the nonlinear effects. So my question is, imagine that we have one of your systems, classical systems interacting that synchronize. Now I am let's say I'm a quantum beast. I know nothing about classical description and but I can easily solve Schrödinger equation. What should I see in my wave functions, eigenvalues, to distinguish between the system that classically is synchronizing or not? What should I see in the wave function or the temporal behavior of the wave function to distinguish these two systems, just looking at the solutions of the Schrödinger equation?
Yeah, sorry, I was muted. If you solve just the time-independent Schrödinger equation, the eigenvalues and eigenstates are identical to the adjacency matrix, for instance, of the appropriate network. So, yeah, the difference comes in, I think, the time-dependent Schrödinger equation. What happens so physically, what happens in the nonlinear coupled oscillator systems is that during each phase cycle, so during each period, the oscillators slow down and speed up, just like a flock of birds would do in order to keep in phase. And so, of course, that's not something that we can imagine in coupled quantum oscillators. But nevertheless, you can solve time-dependent Schrödinger equation for coupled quantum oscillators, and there's quantum mechanical versions that have been looked at for the Kuramoto model.
But would the phases synchronize or what?
Yeah, that's a good question, because I guess the quantum mechanical the studies of the Kuramoto model in the quantum mechanical regime have only been done for very small systems.
Yes, I know. That's my question, exactly.
Right.
Yes.
So
yes.
I guess it's an open question. Yeah, I mean, yeah, I guess you've actually crystallized one of the precise.
Yes, because Ehrenfest's theorem would say that the average should, in a sense, reproduce in some sense the classical behavior. So I should be able to see the synchronization even on the quantum level. But in what sense? Where should I look at?
Right. Well, okay. Well, I mean, you would definitely see it in the von Neumann entropy. You get for the ensemble von Neumann entropy as a function of time. It can go from high to low if you start everything out of phase.
So linearity of quantum mechanics can be deceiving, I would say.
I think you have to have a measurement sneaking in somewhere.
Yes, I think so.
Yeah, you simply get linearity, and there's no way of getting these effects. You don't get synchronization and so on. So there's sneaking somewhere is a measurement.
Yes.
So you consider that part of quantum mechanics, which it is, of course, when you use it, but it's certainly not the Schrödinger equation.
Yes.
Right. Because in my head, I added a dissipation term, which is your measurement, Roger.
Yes, yes. Yeah, right. That's an interaction with a classical system.
Yeah.
Okay, thank you. Yeah.
Thank you. I think the time. Oh, so Roger, I think you will have the last word, as you should.
Well, I had a completely different question. I'm just very intrigued by how much people were talking about electromagnetic effects, and as opposed to just normal nerve propagation and things like that. I remember when I was writing The Emperor's New Mind, trying to learn something about neurophysiology and so on, and I learned about the Huxley-Hodgkin nerve transmission and so on, and I could see no role for the kind of, well, something outside linear quantum mechanics and so on. It seemed to me nothing. Let me check. That was not the point I'm trying to make here. The point I was trying to make is that in as trying to understand these things, I remember being very struck by the fact that you have this fissure between the frontal and parietal lobes, and there are these things called the homunculi, and one is on one side you have the sensory homunculi, and the other side you have the motor ones. And what struck me is how correspondent they were. I mean, something to do with some kind of movement and something to do with some kind of sensory seemed to be pretty well opposite each other. Now, what's the point of that when there are no nerves going across? So they seem to be feeling each other out in some sense, which goes beyond actual nerve propagation. Now, could this be any manifestation of an electromagnetic effect or something? Why? It just seemed to me a great puzzle, which nobody seems to mention, at least I haven't heard them mention it. You just see where these various sensory and motor features to do with moving the hand or moving the leg or whatever it was, and they pretty well opposite each other. Now, what are they doing? What's the point of being opposite when there aren't nerve connections there? So could it be that there is some kind of transmission of information across of a different character, which maybe there's some sort of electromagnetic effect going on? I don't know. Does anybody have a comment to make on this?
Yeah, that's actually been very well studied. It's called ephaptic communication between neurons. So where you have sulci and gyri with columns of neurons on either side, closely opposed, they're well within the distance ranges needed to communicate both magnetically and electrically. And in vitro experiments have shown when you have adjacencies like that, you actually do see the ability of one group of neurons to influence the threshold of firing of the adjacent neurons. So you can depolarize or hyperpolarize. It can work either way. And that was proposed, I think, back in the 50s to be a possible way that we can communicate at speeds well beyond axonal transmission. It could get to the problems raised earlier about why does perception appear to take so long in the brain when in fact our responses are much quicker. So if these ephaptic and electromagnetic fields are contributing to our ability to perceive and respond, that could very well account for the speed differences between the synaptic and axonal transmission versus virtually instantaneous transmission across, small
Okay, well, that would certainly make a lot of sense. I hadn't heard that had been studied that way, but that's very interesting. Yes, thank you.
Bruce, do we know what generates the ephaptic fields?
Yeah, it's the same thing that generates the surface EEG. So you have circuit current flow into the dendrites and back out across the basal dendrites. So in at the apical and out at the basal. And that, just like a regular electronic circuit, generates electromagnetic fields. And it's those fields. This is work done by Jim Miller at UBC many years ago that showed that these electric fields are sufficient to depolarize nearby neurons. And since that time, it's been shown quite clearly that it's not only plausible, but it does happen.
Of course, EEG could be coming from interference patterns in the microtubules inside those apical dendrites. Because as we talked about at the last conference, we don't really know the mechanism of EEG origin, do we?
Yeah, I think we do. As we've studied that in brain slices where there's no thalamus or any other part of the brain except the neocortex, and little mini slices of just neocortical tissue. And it looks like it's circuit dynamics between the upper layers 1, 2, and 3 and the lower layers of 5 and 6 that form reverberating circuits that synchronize vast numbers of neurons, like whole columns of neurons, to so that all their electromagnetic fields align. And that's what we're sensing at the top, at the surface of the brain, is these electric fields from literally thousands of neurons that are synchronously being driven by synaptic currents.
Of course, you remember Cantiello's talk that it was the microtubules generating the gamma synchrony oscillations.
Yeah.
You didn't agree with them, but
yeah,
I don't think we know yet. I'll shut up now. I've talked too much.
No, it's a good comment, and I think we are back to the organization of the brain from the bottom up or top down. I don't know. At any rate, I would like to. We are out of time. I would like to thank all the panelists, all the participants, everybody, for a very exciting event today. Interesting comments, and many happy returns to Sir Roger. We all benefited so much from your wisdom. And thank you for the last comment. I think we'll all think about it and go to Bruce for advice. Thank you all, and see you tomorrow, I guess.
Well, thank you for a fascinating day, yes.
The Royal Swedish Academy of Sciences has today decided to award the 2020 Nobel Prize in Physics with 1/2 to Roger Penrose for the discovery that black hole formation is a robust prediction of the general theory of relativity.